Good morning, everyone, and welcome back to day 2 of the FDA Science Forum, where this year's theme is Advancing Regulatory Science Through Innovation. Before we get started today, I just have a few opening remarks, as most of our continuing education was provided on yesterday. CE will be granted for this event. For more information regarding your preferred sessions, please see fda.gov and the FDA Science Forum webpage. Just as a reminder, our speakers for today are expected to use generic names. If trade names are used for your presentations, those of several companies should be used rather than that of a single supporting company. Also, you're required to disclose to attendees when products or procedures being discussed are off-label and any limitations on that information. So without further ado, I will bring on our first moderator who will open the session for food and cosmetic safety, Dr. Raj Nayak. Raj, the floor is all yours.
Thank you, Sharon. Can you hear me?
Yes, we can hear you.
Okay. So good morning and welcome to FDA Science Forum Session 5 on food and cosmetic safety. It is my great pleasure to introduce the 5 distinguished speakers in this session. We have Dr. Jean Liam from the European Food Safety Authority, Dr. Steve Musser from CIFSAN, Dr. Steve Foley from NCTR, Dr. Luisa Camacho from NCTR, and Dr. Patrick McDermott from CVM. After the presentations, we will have a panel discussion and a Q&A session. If you have questions, please type them in the Q&A box. If you have a question for a particular speaker, please highlight the name of the speaker that you want to ask the question. We will cover as many questions as we can in the panel discussion. Thank you. With that, I would like to introduce our first speaker, Dr. Jean Liam. Dr. Liam is the head of the preparedness team of the Methodology and Scientific Support Unit in EFSA, that's the European Food Safety Authority. Dr. Liam will be sharing his scientific expertise in the areas of methodologies for chemicals in foods with the national and international risk assessment agencies across the globe. Dr. Liam, the floor is yours, sir.
Thank you very much to all of you. I would like really to thank the organizers and particularly a couple of the FDA colleagues with whom we are working very closely together. I would like to start now my presentation, and I'm going to talk to you about ILMIRAC, and I will explain to you what it is. Thank you. Yes, I hope you can see my screen.
Yes, Dr. Liam, if you can put that in the presentation mode, that would be great.
Yes. Hope it's working.
Yes, sir. Thank you.
And you can see the full screen?
Yes, we can.
Okay, that's great. So ILMIRAC is an acronym, a very difficult one, but I've given, let's say, on the bottom of the slide, the full, let's say, name. It's the International Liaison Group for Methods of Risk Assessment for Chemicals in Food. And it is a group which is composed of many, let's say, very knowledgeable institutions around the world. And we have created this platform for methods in chemical risk assessment in foods, let's say in 2018. And it was after some consideration together with many of the institutions that you can see on this acknowledgement slide. Particularly, we had some preparatory meetings before it was created in order to see if there was sufficient interest in creating a platform where we could work together on methods for risk assessment of chemicals in food. You can see it here on the globe, the many agencies in the US, of course, including US FDA. We had quite a lot of discussions already since many years in order to sit together and to see if we can try to work on particular developments in chemical risk assessment, and particularly in the area of food safety. And so at a certain moment, about 10, 15 years ago, we started to collaborate not only with US FDA, but also with many other agencies, as you can see here on this slide. And we were, let's say, connecting to each other mostly in a bilateral way. But after some time, everyone said to each other, I think that everyone has a little bit the same problem that they would like to work on together, if possible, with other institutions. And that's what we tried to do, and it was particularly then leading to, let's say, the agreement that we would start with ILMIRAC with about 20 members and observers in the beginning, and it has grown until now, until 29. And we are still expecting that particular institutions would join us. Hopefully, one of the continents which is still lacking from the continents in the globe is Africa, which is working on creating food safety organizations across that continent. And we hope to have also Africa joining and engaging with us in ILMIRAC in the near future. So today's presentation, I would like to introduce you, of course, to the objectives for creating ILMIRAC and how we were starting to work together, and particularly with defining the sort of terms of reference that we are making use of in order to let's say, develop targets for us that we would, let's say, like to work on all together. And then I will show you a little bit in a nutshell all the activities that we have been doing so far. It is not that we were, let's say, talking to each other every day. No, it was just keeping it practical and trying to organize meetings, particularly conference calls during the year, in order to keep track of what is happening in the area of food safety risk assessment, and particularly in the area of development of methodologies and their implementation. And of course, from time to time, it is important that you also try to identify if the organizations involved in ILMIRAC were considering the activities useful, and if there was anything that we should improve in terms of collaborating together in this platform. And of course, I would like to end a little bit with lessons learned, in particularly what the benefits are that we created this international liaison group, and what is the outlook. So going to the next slide, you see here, this is in fact a sort of laying down the objectives that we have included in the terms of reference for ILMIRAC. It was in fact built upon, let's say, the interest of many of the organizations that were starting in 2018 to think about, let us sit together, because everyone is probably working on the same kind of topics in methodologies for risk assessment. And it would be quite useful to hear in one go from all of us around the table what the ongoing activities are, and particularly if there is a possibility for working together, joining forces, but also avoiding duplications of effort, making use of what everyone has been doing so far, and sharing the information which is already made available or published as far as possible. So the support of that one of the objectives was to support the development of new risk assessment methods for chemicals in food. There were lots of agencies already working on how can we improve the chemical risk assessment, how can we speed it up, how can we make use of new technologies and tools that we could take on board, and that we can also harmonize how we are using these tools and instruments in the chemical risk assessment. improving the consistency of the methods used for risk assessment. Of course, if you are dealing with the development of models or you're developing all kind of databases, it would be good that you at least can share it also with others, but in such a way that they understand how you need to use it and how you also need to interpret all the information that is put in databases. So more specifically, mapping activities, ongoing activities in the different agencies, particularly then related to developing, validating, and implementing methodologies, particularly also working together with the observers, the international organizations who are really dealing with international standard setting. And here we, of course, have to talk about the World Health Organization, the Food and Agriculture Organization, and OECD, the Organization for Economical Cooperation and Development. And these international organizations are, of course, working together with all the member states under the United Nations, which are dealing with, let's say, risk assessment and with all kind of other topics. Then to propose action plans for implementing and particularly for harmonization of the methodologies, to facilitate harmonization through organization of joint events, joint projects, and also exchanging staff where possible, learning from each other, and also looking at how it is being done and being used in practice. Then assist in identification of research needs and trying to promote the application of new science in risk assessment practice. As you can see here, of course, if we talk about new methods for chemical risk assessment, it can be developed by different types of sources. It's in academia, research and development. We have, of course, many institutions which are dealing with the validation of methodologies. And of course, there should also be some discussion about the validity, about, let's say, how to use it. And there should be some review independently so that there was, let's say, a possibility to gain trust and confidence in increasing the acceptance of particular methodologies, which could be very useful in the regulatory frameworks. And this is particularly important for those institutions which have gathered together in ILMIRAC. The working arrangements that we agreed upon in the beginning of 2018 was, let's say, sit together every month or every quarter, but let's at least sit together with those organizations who are really having a sort of formal or official mandate for food safety risk assessment, governmental, intergovernmental food safety organizations. We had agreed, as was also already done in other liaison groups, to have at least one of the organizations offering the possibility to provide a technical secretariat. And of course, this is a secretariat which would then be coordinating, planning, organizing meetings, to draft agendas, keep the things moving, and also track actions where needed. And that technical secretariat was agreed to have it rotated among the participants in ILMIRAC every 3 years. The type of contributions, exchange of information on ongoing and planned activities, as already said, but particularly also if there was already a methodology described by the agencies, it would be very nice that we could share these guidance documents that we are maybe everywhere using for describing how we are applying the methodologies for risk assessment in practice. Also, of course, the possibility to exchange experts. If there is a particular center or a particular agency with, let's say, particular expertise in benchmark those response evaluations, we would, let's say, give that opportunity to institutions interested to second experts for a couple of months to that particular institution. So the ILMIRAC activities, and then, of course, it was also to be considered, if needed, to create ad hoc working groups that would be dedicating its focus on particular domains of the methodologies or the tools that we want to implement in risk assessment. Meetings, 2 to 3 conference calls a year, And we were, let's say, in the beginning agreeing, well, let's at least also meet physically in order to know each other better, to know also, let's say, the different institutions in more detail. And that was the plan to have that every 2 years. Confidentiality arrangements were made, and particularly it was during the in the meetings for the meetings of ILMIRAC, it was very much important that everyone knew how to handle sharing documents, documents that would be shared. You have always to know the status of that document. If it is a draft which can be shared with others, if it is a draft that is, let's say, partly confidential, of course, we need to be very careful that we, let's say, explain how to use particular documents that are shared within the international liaison group. This is an overview of the activities. It's one slide, but I can tell you it's a lot of, let's say, teleconferences that we have been holding in the 6-year period almost. We will have the next one, the 15th teleconference will be happening tomorrow. It will be, let's say, from 12 to 2 o'clock at lunchtime in Italy. And as you may imagine, if you would, let's say, have a closer look into which organizations were taking part in these conference calls, some people were still in pyjama or were going to have a pyjama at the end of the meeting. And that is particularly because we were, let's say, sharing and having these conference calls with people in South America, the US, Canada, until New Zealand and Australia, with, of course, lots of hours of time difference in between. So for that reason, we try to have a sort of agreement. Okay, let's put it somewhere in the middle so that everyone can take part in the meetings, and we would then also limit the meeting to 2 hours in total, where we would then effectively, as effectively possible, discuss a couple of topics. And that was, of course, together with all the participants, we were agreeing on which topics we would then focus the agenda. And then, of course, what we try to do is upcoming publications, if there would be particular sensitive methodology, which would be very interesting for everyone, that if that would be coming out at a certain time point, we would alert and announce it to the participants so that they can also take that on board in the planning of their activities. What is also very important and which has been useful all the time was to share lessons learned in particular, and particularly in the application of methodologies. Everyone knows that you sometimes make mistakes in directions taken for the development of methodologies or tools. And that is particularly important for everyone to know. So for that reason, we also were quite open in terms of don't do that, because it would lead to particular, let's say, limitations, for instance, in sharing the information with others. So, of course, if there would be, let's say, templates that everyone could use, which would be ensuring that people can always open it and can access it, that's very important. And then the special working groups. We identified that that was in 2020, after the midterm evaluation that you can see on the lower part of the slide. We had evaluated how far are we? Is it really, let's say, making sense to keep ILMIRAC alive? Is it giving the added value that that the organizations were expecting? And on the basis of that evaluation, we came to the conclusion, among other things, that we would also enhance the collaboration by making specific working groups. And one of the working groups is very active at the moment. It's in the area of new approach methodologies, which is co-chaired by Suzanne Fitzpatrick, your colleague in US FDA, together with Jose Tarazona, which is a former colleague of mine, but also now working at the CNSA organization in Madrid. But this is a very active working group where we are really, let's say, dealing with a topic which is of interest for many, many of the participants in ILMIRAC. So that was one of the reasons that that we concluded, let's do that, because this would enhance a little bit more the sharing of expertise, sharing and joining forces in particular activities. And that is in particularly, let's say, the added value that was seen by everyone in ILMIRAC. This is just to show you a couple of slides that we were using in the evaluation of the so-called ILMIRAC survey, where we evaluated the added value of ILMIRAC. And of course, one of the questions that you would ask the organizations is it useful for you? And particularly, there were a couple of topics identified where they said, yes, it was very useful. And you can see that on the left side. It's particularly information exchange on methodologies, of course. Directions taken for new guidelines that were, let's say, taken up on board in development, ongoing plant activities. But we, of course, also asked what is not yet, let's say, useful or as sufficiently useful as you were expecting. And that was sometimes with topics which were not really shared, having a shared interest by everyone, but certainly they were sometimes indicating, listen, if we are going to do more development and implementation of harmonized methodologies, that would be, let's say, certainly very useful for all the participants or for more participants. And of course, also what we were not, let's say, sufficiently doing was to identify gaps and directions that could be taken on board by the international standardization organizations like WHO, FAO, and OECD. And this would then be enhanced in the period afterwards. This is a little bit, well, the mode of presentation that we were using for the evaluation. Here you see a couple of topics which were identified as a topic for consideration by ILMIRAC. And you can see on the left side, maybe it's a little bit little for you to see, but there are a couple of topics which were such as chemical mixtures, such as new approach methodologies based on non-animal testing, making also use of the possibility to keep all the organizations informed about ongoing developments in priorities, what the organizations are giving priority in the area of methodology development. This is, let's say, another, let's say, slide, which is in fact identifying what they were considering as topics useful or not useful, and sometimes also identifying why it was not considered useful. And that was, of course, also a sort of lesson learned that we took on board in, let's say, developing the agendas for the conference calls that came afterwards. Proposed areas for improvement. I will come back to that in, yes, this slide. In fact, what we concluded after this ILMIRAC survey in 2020, everyone was still enthusiastic to continue participation. And I can tell you the enthusiasm was also enhanced by let's say, an increased number of organizations that were asking to take part. And we are now with 29 organizations, and they all wanted to continue participating in the ILMIRAC meetings for another 3 years. With the same frequency, because of course every organization is having its own program of work, and of course you cannot require too much that let's say we are having too many meetings, and keep it, let's say, feasible for everyone. And of course, if it would be every 3-4 months, let's say, particular priority settings would not be drastically changing. So realizing this, we kept the frequency of conference calls at 3. Unfortunately, the COVID period was limiting us in organizing face-to-face meetings. But luckily this year in September, we are inviting now all the participants of ILMIRAC to come to Parma in Italy and to have a meeting, let's say, of one day in connection also with another event, which of the global coalition that you all probably know, the Global Coalition for Regulatory Science Research. And that is, let's say, a week of in September, where we have a couple of events connected to each other, and which is allowing people to attend these meetings. So very pleased that we can invite all the people to participate in that face-to-face meeting in Parma in Italy. And I hope that we have a good time, of course. And of course, we continued, as F said, to provide a technical secretariat. And we are now asking again to the organizations if they would like to take over, because in principle, it should be every 3 years that we are rotating. And we have those improvements that I was talking about already, the working group that we have created on new approach methodologies, which is a sort of online working group, and for which we have very good discussions about this particular topic. We also wanted to facilitate that all the meeting documents would become better accessible for everyone. And so we are now making use of Microsoft Teams, which is a package that you can use. It is, let's say, sometimes working well and sometimes not working well, but particularly this is also used for sharing information in particular folders, and everyone can go back in time until even the first meeting of 2018 to go back in time to see what kind of presentations we had, the materials, including the agendas, the meeting reports that we are making after each of these conference calls. And this was also to facilitate accessibility and, let's say, looking back into what was, let's say, leading to a particular activity that is now ongoing. And then, of course, also trying to make use of particular tools, particularly also, of course, for the non-English speaking members, and they were really asking if it would be possible to record the meetings, as we are now also doing in this FDA Science Forum, and to implement tools that could be useful to have a good understanding of what we have been discussing during the conference calls. The benefits of ILMIRAC, yes, we have certainly always targeted that we would like to have consistent approaches across the globe, aligning risk assessment methodologies that we are developing, taking on board lessons learned from others, and also even joining forces in order to improve particular aspects of chemical methodologies for the risk assessment. And certainly also, of course, making use of already the guidance documents, and particularly we all know that there are particular textbooks issued by FAO, WHO, OECD has a lot of guidance documents issued. And one of the examples that has been given here is the Environmental Health Criteria 240, which is issued by FAO and WHO together. And this is covering lots of principles and methodologies for risk assessment of chemicals in food, and which is being used by all the organizations that we have in ILMIRAC. Another benefit, of course, working together, looking at each other, what we are doing, trying to see opportunities for joining forces, sharing expertise. This is particularly, let's say, really leading to synergism, and particularly the NAMS working group that we now have, the new approach methodologies, where we look at the use of new approach methodologies without animal testing. And IATA, which is the integrated assessment, integrated approaches for testing and assessment. And of course, what we really would like to have is also that we can improve confidence of consumers, stakeholders in the risk assessment methodologies and approaches and data that we use in the risk assessment practice, and particularly through international harmonization and collaboration. This is an important aspect. We are, let's say, even strengthening, let's say, the work in order to share the information that we have in the methodology development, and particularly not only to improve the confidence of consumers and stakeholders, but also our own experts sometimes need to get convinced that you can make use of new approaches in chemical risk assessment. And that has become an important topic in the development of new approaches in the food safety area. And the benefit for any given scientific risk assessment agency, of course, you have all kind of topics that we are considering in ILMIRAC, and you can see that on the right side. Any plans that you have in order to start an activity is useful for sharing, and particularly sometimes you're even making use of the possibility to have a consultation more widely, more publicly on a particular mandate that we are going to take on board in EFSA or in another organization like FDA and also, of course, FAO, WHO. They are also doing that in a similar way. Identifying new challenges, new themes, new models and tools to ensure that people know that particular instruments can be useful and are already available, sometimes in an open science forum, and identifying also risk assessment partners for particular methodologies that a given agency would like to work on. And of course, what I already said a couple of times, international harmonization, making it more consistent in order to avoid that there are unnecessary debates about the differences in methodologies between different agencies across the globe. I would like to end my presentation with acknowledging, of course, the colleagues that I am very much working together with: Suzanne Fitzpatrick, Jason Ongst, Sherry Dennis, Christy Muldoon-Jacobs, and also, of course, Ashley, with whom I was in contact in order to organize this presentation. And all these EFSA colleagues that I'm mentioning here, of course, we are working together with all colleagues across the agency to put things on the table of ILMIRAC. And you can see it's covering all the domains that we have in EFSA, all the domains in the area of food safety. Would like to thank you all for your attention, and I'm happy to answer any question that you may have. Thank you.
Thank you, Dr. Liam, for that wonderful presentation. It's very gratifying to know that FDA is partnering with ILMIRAC, and we are closely working with the European Food Safety Agency on the risk assessment of chemicals. We'll hold the questions for a panel discussion.
Okay.
Thank you for your time, sir.
You're welcome.
So our next speaker is Dr. Steve Musser. Dr. Musser is the Deputy Center Director for Scientific Operations at CIFSEN, and he will be sharing the progress and needs for new alternative methodologies in CIFSEN's regulatory mission. Dr. Musser, please go ahead.
Good morning, and thank you, Raj, for that nice introduction. I certainly appreciate it. It's very nice to be here speaking again at this meeting. I believe I've had an opportunity to attend and speak at most of them, if not all of them. I'd like to talk to you all this morning a little bit about alternative methods, new alternative methods. And for those of you unfamiliar with this, this covers broadly non-animal methods or methods intended to replace animals in testing regimes for safety and information on safety data for foods. And it's a very, as you might imagine, a very large area. What I'd like to talk about today is primarily those methods that would cover very large areas as opposed to specific targets like measuring bot toxin versus measuring some unknown chemical in a system like developmental neurotox, which is what I'd like to talk about today. So FDA has a long-standing interest in this particular area. We have a commitment to develop and use new technologies in general and to evaluate, predict the safety and effectiveness and reliable manufacture of regulated products in all the areas that FDA works in. And we recognize that these new technologies may help bring FDA regulated products to market faster with improved efficiency or prevent products with increased toxicologic risk for reaching the market. And it's this last point that I'd like to talk about, and that's particularly related to foods and how we use these new methods to look at the safety of chemicals that are either proposed to be entering the food supply or are already in the food supply, and we'd like to evaluate their risk or have some understanding of their safety without using animals as one of the protocols. So the basis for the NAMS investment so far has been to replace, reduce, or refine animal testing. The agency receives quite a bit of requests to stop using animal testing from a number of NGOs. Also in the cosmetics and a number of other areas throughout the world, animal testing is banned for looking at safety of cosmetics and for many other chemicals being reviewed and used in the human foods arena. So there's also a real promise of faster and more efficient identification of toxicity and associated pathways. So in this way, we're looking at what particular pathway may be involved. And if we know that particular pathways are involved in toxicity, or we can point to specific areas that are in toxicity, then we can really start to look at these things faster. The third, and this is very important for foods, is its better modeling of human toxicity. So this has been a problem for toxicologists and toxicology studies throughout the world for decades. When a particular chemical is looked at in an animal, but that animal does not properly reflect the human condition. And so toxicity is observed, but for whatever reason, based on genetics or just that animal model, that toxicity would never be observed in humans simply because of the systematic pathway that it went through. And lastly, there's a promise of lower overall costs. Although I will tell you, based on the models that we have looked at so far, these are very expensive in many cases, especially if you're looking at things like developmental neurotox or particular chip studies where you're looking at like gut on a chip or liver on a chip, or combining chips together or things together. I mean, you can get over $100,000 for a small study on one chemical extremely quickly. And maybe not have all the information you need. So that's just one part of it, and something to consider as we move to regulatory use of these. So as Dr. Liam mentioned, we have a number of committees, both domestically and international. ICFAM is probably the largest committee where FDA participates in worldwide. That work group on alternative methods is extremely active. There's a lot of work in this area on new test method evaluation and acceptance. It includes also the US groups. There's also ICFAM, which is more of a domestic federal partnership between US government agencies. And more recently, internationally, the European Union has just developed a partnership for the assessment of risks for chemicals. This is a large program, a 5-year program started last year. I believe it's a 400 million euro project just to look at assessing risk using these non-animal new approach methods. We're proud to be have a membership in this particular group, and look forward to seeing how this works out. And then also within the FDA, there is an internal working group on the use of non-animal methods. So it's a very big deal. It's a very big interest, and there's quite a lot to be done in this area. Okay, so here is the game. Traditionally, we use a chemical hazard or a chemical that we would like to evaluate the hazard of. We do an animal study, and then from that data developed in that study, we try to estimate human risk. That's the gold standard way of doing these evaluations. And the goal currently is to actually, well, not currently, but in the future, is to take the same path, but instead of using animals, use in vitro methods combined with in silico methods to skip the animal toxicity and move directly towards estimating human risk. Now, probably where we are now is using the in vitro and in silico methods to help design better animal toxicity studies. I think we're still quite a long ways from just leaping directly to estimating human risk from these methods. And by that, I mean, we could use these methods to help us design better animal tox studies. So let's say, for example, that we need a particular type of rat or a particular modified mouse that would be a better estimate of the type of toxicity because we know the pathway or we know the mechanism of toxicity or the proposed mechanism of toxicity or even the targets used in the toxicity. And so we would hope by using these methods currently that we could develop better animal toxicity data, better animal testing, more targeted data that yields results that we can immediately use. So that's the goal currently, and ultimately to replace animals if possible. Okay, so that's the easy part of it. It's easy to say that. It turns out that there are lots of these methods. There's lots of in vitro methods. There's lots of in silico methods, SAR types of approaches, structure activity relationship approaches to looking at this. Many in vitro models, from cells to 3D models to chips. There's also alternative organisms, C. elegans worms, zebrafish, flies, planaria. There's other animals, alternative organisms that can be used. I'd say based on some of the most recent OECD work, probably zebrafish in this area are going to be the most forthcoming or advanced, as they've taken the most leaps in terms of acceptance and use in some of these testing. And I'll tell you why in just a minute. But we have to kind of go through all of these models and look and see how they would fit into the regulatory paradigm. So as we move towards regulatory use, as regulators, we have a lot of questions. And I can tell you that these methods, while there are many of them out there, they have not, many of them, most of them, have not reached a level of maturity that we might use in regulatory use. And this would be, again, for the more complex chemical analysis as opposed to looking at a target for, say, botulinum toxin, where we know the targets and we know what the reaction would look like, and we can estimate the data we need from a very targeted assay. These assays that I'm talking about would be looking at developmental neurotox or liver toxicity, kidney toxicity, cardiovascular disease. These are much more difficult and are not really, in many cases, even qualified. When you ask manufacturers, how do you look at your cell lines? Are they the same? How do you manage to look to see if they give you the same results? Many don't know. Reagents are switched regularly, so a different reagent is just substituted as it becomes available. There's not a lot of knowledge about how those reagents are used, and supplies in general, even types of petri dishes that are used, or plastics, or the types of analysis that are done are not standardized. And there's also how much of this is reproducible? In many cases, these assays are only available in one laboratory, and it might be graduate students or technicians running it, and they've not been reproduced outside of that one particular data. We also need to know a lot more about what test compounds have been assessed. I think there's only a handful of tests that have really been assessed. There's a platform on developmental neurotox that was just recently approved by OECD. It's a 17 assay test, 17 different assays in the test system. And in that, they've looked at both what are positives and what are negatives. What group of chemicals give a positive reaction and what group of chemicals have no reaction in the test. That is really the only one so far. And I would say zebrafish are probably the next test system that has had very similar types of work done. And zebrafish in many cases have actually been evaluated now in multi-lab studies. Interestingly enough, not always getting the same result. So there's a recent round robin of four laboratories, and two of the laboratories got, for the most part, the same results, but two of the others did not. So still a lot of work to do there, obviously. The other question that we would have is, when you define these compounds, what's their applicability domain? Which ones would work and which ones wouldn't? So if we were to look at polyfluorinated compounds that stick to all the plastics and aren't really soluble in everything, how would you use them? Are they even soluble in the material that's being tested, or do you know that they're even being exposed to the cells? This is a big problem, something that we would need to understand and to use. We also need to define the limits of the assay. So we're looking at test data. How much before you see a result? What's the exposure? How much sensitivity is there for the test? Is it very narrow, where you can only dose a specifically small amount? Or is it very wide, where you can see very, very large differences in exposure assessment? And is it specific? Do we know what target is actually being impacted? And nearly lastly, we need to think about relevance. Is it relevant to the human condition? Is it relevant to what we are looking at or would like to look at for a chemical evaluation in humans? What endpoints are being measured? Are they predicted of in vivo effects? So, in some cases, we would need to consider, are they even bioavailable? Are these chemicals, while they're put into a system of cells, for example, would those cells in a typical in vivo system even be exposed to these chemicals or their metabolites? These are all things that we would need to consider, haven't been considered, or being considered, but are still not quite ready yet. Something that's really, really very interesting from a foods perspective is that, with very few exceptions, many of the things we're looking at are chronic exposure. So we're looking at exposure over a lifetime of an organism, not necessarily acute toxicity. We do have cases, for example, in some dietary supplements and where you might see an acute toxicity, but in many cases with measuring chemical toxicity or evaluating chemical safety, we're looking at chronic exposure. So how do you do those tests? How do you look at the data from tests where you're looking at chronic exposure? Still something we would need to have developed before we could really jump into using these tests regularly. And lastly, is it translatable to humans? So if you're looking at a zebrafish, are your endpoints relevant to humans? You may see a result, but is it something related to fish, or is it something that also translates to humans? So lastly, and this is something that we're really going to need to figure out how to use, is you have these in vitro models in the yellow box up at the top. You also have in silico modeling, alternative organisms, artificial intelligence that can be used here, and exposure data, PK, ADME types of data. How do we integrate all that data and pull it into one place? How do we use it for comprehensive risk modeling? Essentially, we've taken the organism apart so that we're not using whole animals. And in many ways, we now have to put it back together and see how all those pathways are integrated, how we would use them, and how we would model the whole organism based on the data we've obtained from separate pieces and parts of the analysis of the data of the safety. So lastly, there is huge promise in this area. I know I've highlighted a number of areas that need to be developed and that we need more information on. But there's a lot to be going on. There's a lot of really interesting, useful work that's being done. I can't stress to you how important this is going to be going forward, but there needs to be a greater emphasis on qualification or validation of these tests in general for regulatory use. There needs to be much more collaboration and comparison across laboratories. There's only been one or two or three types of comparisons across laboratories for many of these tests. And more work on data integration. There needs to be a lot more from the start on how are we going to take these evaluations and integrate them from in silico, in vitro, non-animal model organism types of approaches. So it's a lot of work to be done, but a lot of very promising positive information that can come out of these tests. So with that, I will thank you for your attention. I appreciate the opportunity to speak here this morning and look forward to answering questions at the roundtable discussion. Thank you.
Thank you, Dr. Musser, for that great presentation. And obviously, there are some challenges for the regulatory decision-making, as you mentioned, especially from the qualification, the validation of these methods for testing compounds, and lastly, to how do you translate that into humans? So we are getting some really good questions, especially for the new alternative methodologies that I'll share with you. But just a reminder, just feel free to send your questions in the Q&A box. Thank you, Dr. Musser.
Thank you.
Our next speaker is Dr. Stephen Foley. Dr. Foley is the director in the Division of Microbiology at NCTR. Dr. Foley will provide an update on the studies to assess the virulence of enteric foodborne pathogens. Dr. Foley, please go ahead.
All right. Thank you, Dr. Nayak, for the opportunity to discuss some of our work on virulence in these foodborne pathogens. And so we look at data and estimates from the CDC. We see that there are the bacterial foodborne pathogens cause a number of illnesses. And when we look at those enteric organisms, those highlighted here in red, we see that among the most common causes of bacterial foodborne illness, enteric organisms make up a large chunk of those. For the talk today, I'm going to largely focus on Salmonella as kind of a model system for that. And Salmonella, again, one leading cause of bacterial foodborne illness, estimated over 1 million infections each year, leading to about 20,000 hospitalizations and nearly 400 deaths. And when we look at kind of historically serotypes such as Typhimurium, Enteritis, Newport, Heidelberg, and have been some of the most common causes, over 100,000 cases in the period highlighted here. Most of those come from stool samples, so the typical gastroenteritis, diarrhea, vomiting, nausea, those types of stuff. We do see a percentage of those that come from more invasive sources like blood, spinal fluid, joints, urine, wounds, abscesses, and those. And so one of the things that we've been interested in is trying to understand, what are some of the factors that contribute to some of this diversity of virulence in there, and trying to understand what makes Salmonella an important foodborne pathogen. So there's been a lot of work on Salmonella virulence, and in order to cause infection, Salmonella need to be ingested with the food, get through the low pH of the stomach, and then they interact with the intestinal tract, with the microbiota, the intestinal epithelial cells. A number of Salmonella have virulence factors that will lyse or kill competing bacteria and allow for colonization. And then once colonized, then they express what's called a type 3 secretion system that allow them to get into cells and another secretion system to survive. And in the different cell types, they can move from the luminal side to the basal side, where they can then interact with immune cells like macrophages and and dendritic cells and get taken up. And these are often mobile and then can be moved to other parts of the body and can potentially lead to spread to different areas of the body in there. And so if these more severe cases where you have the blood, spinal fluid, those types of things, are those cases where antimicrobial use is often indicated for treatment. And so antimicrobial resistance becomes a concern in those. And that's not going to be the main focus of the talk here in there. But a lot of these resistance elements, as well as virulence factors, are located on plasmids, or these extrachromosomal DNA elements that are often mobile, and through a process called conjugation can move from a donor strain into a recipient strain, and then that strain now would pick up the either the antimicrobial resistance or virulence factors that are carried on those plasmids. And our research group, we've been doing a lot of work with sequencing of plasmids and looking at sequenced plasmids that are available publicly. And some of the major ones with salmonella and other enteric bacteria are those that fall into what we call incompatibility groups, INC HI2, I1, FIB, and AC. These are common resistance plasmids. One of the concerning things with examples like the INC FIB is that in addition to these antimicrobial resistance elements, they also carry virulence operons that may be associated with iron acquisition or manganese acquisition, or the bacterial toxins that may lead to improved colonization factors. And these are often then carried with antimicrobial resistance elements. So you get kind of that double whammy, if you will, from these particular plasmids. And so I'm looking at those plasmids like the INC I1 and FIB quite a bit, trying to understand their roles in virulence. And again, several of those carry those bacteriocins or colicins that can lyse competing bacteria and help maybe with the colonization part. The INC FIB and some of the others carry multiple iron and manganese acquisition operons. And one of the things that the innate immune system does is it chelates iron and other cofactors away from the bacteria. The bacteria need those. And so in kind of a counteroffensive, the bacteria that acquire these are able to go out and get that iron, bring it back in. And we see this as in a lot of the organisms that are associated with avian sources, both things like Salmonella serotype Kentucky, as well as a lot of the avian pathogenic E. coli. And they seem to play a role in virulence or pathogenicity in these organisms, these species like poultry. Another one that we've noticed, we've saw in some of our sequencing studies were these INC X4 plasmids, and these encode what's called the VERB-D4 type 4 secretion system. And we'll talk a little bit more about that in detail, but these have been found in some outbreak strains of Salmonella that have caused a number of human illnesses. So what do these virulence factors look like when we're studying those? So we were doing some initial conjugation studies where we did what's called plate mating, where we took our recipient that lacked the plasmid and struck that on the vertical, and then took our donor strains that had plasmids and cross-streaked those across. And the goal or the plan was to, where those intersected, get the mixture of bacteria and see if the plasmids transfer. Well, when we were working on those, one of the things we noticed that a lot of our strains was that there was a zone of clearing here. And so something being produced by the donors were killing the recipient. And when we looked at the plasmid, the sequence of these strains, those are often those carry the colicins, both either on the INC FIB or the INC I1 plasmids. Again, I mentioned the FIB plasmids also carry iron acquisition factors. And when we've done looking at studies in survival and persistence in intestinal epithelial cells, the strains that have those plasmids are much able to survive under iron limiting conditions compared to those lacking the strains. This is work that was done by BJ Kajanchi, who's now worked with us now as at CIFSAN. And so these plasmids do seem to be playing a role in survival under low iron conditions. And we looked at gene expression then, and those genes that are associated with iron acquisition were significantly upregulated in these iron depleted conditions versus the control or the iron-rich environments. So under those low iron conditions, they're being activated and are playing important roles in ability to get iron for these bacteria. Those INC X4, that VERB-D4 type 4 secretion systems, they're encoded on these smaller type plasmids in there, but they encode these type 4 secretion systems, and they allow for DNA or potentially proteins to be moved from the bacterial strain into either a host cell or another bacteria where they may elicit a function that can alter the host or other bacteria. And what we saw with strains that carried those INC X4 plasmids, when we looked at survival under in macrophages, we had the control that were unaffected. This strain is where doesn't contain the INK-X4 plasmid, and these other 4 do. This is the same strain with the plasmid added to it. And what we saw after an hour is that macrophages ended up being killed in there. And that's important because remember back to the cartoon before, those macrophages are mobile a lot of times. And so they could get the bacteria taken up and moved to other sources in the body. And then if they're killed and mice and bacteria are still viable, they can then spread to other parts of the body, which is not a concern, or which is a significant concern, excuse me. One of the challenges, though, with studying plasmid factors is that there's not a lot of great tools to study those. And so Deresiy Gudeta, who's in our lab, has been developing some neat tools to be able to knock out genes on plasmids and get around some of the issues that are there using different colorimetric screening, so either pink-white screening or blue-white screening, so we can look at, like in this case, where there's a white colony in the field of pink colonies. Those are the ones that likely have the genotype we're looking at, and so it makes a much more efficient selection process. So we've used that. Deresiy's used that to knock out portions of that VERB-V4 type 4 secretion system where the Aerovac and Operon in there. Now we're able to go in and look at what is their role on function, say under iron limited conditions or survival on macrophages, and those are studies that we've got ongoing at the moment. Another challenge with plasmids is, we may want to look at individual genes on those, but we also may want to look at what's the function of the whole plasmid in there, are all the genes on there. And there's a process called plasmid curing where you get rid of those plasmids, and that's been a real challenge in there because plasmids have what are called toxin antitoxin systems on those, or host addiction systems, and they encode a long-acting toxin and a short-acting antitoxin. So if the bacteria lose the plasmid, the toxin sticks around, but they lose that antitoxin and the bacteria are killed. And so to try to get around that for curing, Deresiy created this antitoxin expressing vector that with multiple antitoxin genes associated in there. And so you can then cure the plasmid, and this one stays around in there with the antitoxin genes, and you can kind of wash out the toxin. And this is temperature sensitive, so then you alter the growth temperature and you can then lose this plasmid as well too. And so we were able to show plasmid curing with that for a number of the different plasmids types. So these tools are now available, and we're working on trying to optimize those. Another area that we're working on is trying to develop improved bioinformatics tools to utilize that whole genome sequencing that's out there. And so we've developed a Salmonella virulence gene database, as well as a E. coli virulence gene database. And the Salmonella one's a bit more advanced at the moment. So we looked at sequence strains as well as the literature and came up with a comprehensive database work being done by Dr. Jing Han in our group. And what this has allowed us to do is take whole genome sequencing, upload those into the database, and then you get a readout with kind of a blast search of the different genes that are present, their identity, and so you can look at gene conservation, those types of stuff. And another tool allows us to upload multiple strains, whole genome sequences in, and you get then a presence-absence matrix in there, and so you can start to compare virulence gene profiles with programs such as bionumerics or others that are out there for phylogenetic analyses. And so we've done that with several thousand Salmonella strains. And what we saw when we did the principal component type analysis is that they often grouped out based on serotype due to the virulence profiles in there. So this is Salmonella typhi, which is not generally considered a foodborne pathogen, but it clusters outside of the others because it contains a number of unique Salmonella pathogenicity islands, whereas the foodborne one generally lack these. There are some serotypes that contain SPI11, for example, but most foodborne ones lack those. And so what that's done is it allowed us to work with other groups like Heather Tate at CVM to analyze different serotypes. This is some work with Salmonella Kentucky that was done. And so Salmonella Kentucky is unique in that it's very common in chickens, but it's a relatively rare cause of human infections. human infections generally fall in this group called S sequence type 198. And so when we looked at the different virulence profiles, the sequence 198, type 198 clustered outside of the main group. And we looked at the genes that were present. This TCF operon, for example, was one of the things that the 198 had, the others did not. And so we can start to drill down on that as well. And so one of the things, just a quick in conclusion, that virulence in salmonella and other enteric organisms is quite complex and multiple layered in each of the different steps in that and pathogenesis. And so trying to understand that's been quite complex. Then you throw in plasmids, which are mobile and can move from one bacteria to another, and that also creates additional challenges as well. And the concern there is the co-location of virulence and antimicrobial resistance genes as well too. And so we've been working on trying to develop tools to look at individual gene function, look at plasmid function, as well as databases to utilize the wealth of and other tools to utilize the wealth of whole genome sequencing available. So I'll stop there. I thank everybody.
Thank you, Dr. Foley, for that wonderful presentation. And I agree, plasmids and incompatibility replicons do play a major role when it comes to salmonella fitness. Our next speaker is Dr. Luisa Camacho. Dr. Camacho is the Deputy Director in the Division of Biochemical Toxicology at NCTR. And Dr. Camacho will provide an update on NCTR and the Office of Cosmetics and Colors, the OCAC, collaborative efforts as they support the cosmetic safety regulation evaluation. Dr. Camacho, please go ahead.
Thank you very much, Raj. Good morning, everyone. As Raj mentioned, my name is Luisa Camacho. I'm the Deputy Director of the Division of Biochemical Toxicology at the National Center for Toxicological Research, or NCTR. And I'm very grateful for the kind invitation by the organizing committee to provide me with an opportunity to share with you some of the work that we have been conducting at NCTR in collaboration with the Office of Cosmetics and Colors in support of FDA's efforts towards cosmetic safety evaluation. So this is just our standard disclaimer. So I thought I'll just start my presentation by briefly introducing to you NCTR, since many of you may not be familiar with this FDA center. So NCTR is the research center at the US Food and Drug Administration, or FDA. And unlike the other centers, we do not have regulatory authority. So rather, our mission is to conduct research to address data needs identified by the FDA product centers, as well as to develop and evaluate new testing tools that may inform or have the potential to support FDA's regulatory decision-making. And so you heard just a presentation by my colleague, Steve Foley, with great data to illustrate this mission. And it's also within this mission that we collaborate with the Office of Cosmetics and Colors, or OCAC, to contribute to FDA's cosmetic safety evaluation. Our current collaborations with OCAC can be grouped loosely into two categories, one that deals with tattoo ink research and one that focuses on in vitro skin permeation testing. And in the interest of time, I will not have be able to cover all these studies, but I'll just provide a few examples of these studies. And towards these efforts, we contribute with different expertise, including microbiology, chemistry, pathology, as well as the conduct of in vivo bioassays and in vitro testing. So I'll start by sharing some of the work that my colleagues at the Division of Microbiology have been conducting for a few years now, surveying tattoo inks and tattoo-related products sold in the US market for microbial contamination. So why are we conducting this work? So as most of you know, tattoo inks are popular cosmetics, and there's an estimated 20 to 30% of the US population that have at least one tattoo. And actually many of these people have more than one tattoo. However, infection is a primary complication that can result from tattooing, either because issues are related to the healing process or because of the use of unsterile equipment and needles. But this infection could also be because the tattoo inks themselves may be microbiologically contaminated. And so the goal of this research program is to conduct surveys of tattoo inks and related products sold in the US market for microbiological contamination. So this work is being led by my colleagues Peter Kim and Dr. Peter Kim and Dr. Alex Kwan, and they have conducted several rounds of surveys for a total of 197 products sold in the US markets, and found that about 1/3 of these products actually showed microbial contamination. And these included both aerobic and anaerobic bacteria, and some of them were clinically relevant. They've published several papers on their work, and so I welcome you. I'm listing them here, and I welcome you to read more if you want to learn about this important work. Just to give you an idea of their findings, so as I mentioned, about 1/3 of the products surveyed, they had microbial contamination. And interestingly, this finding did not seem to relate to the sterilization claim in the label of these products, with about 1/3 of the products that were labeled as sterile still showing microbial contamination, which is a rate similar to those that were not labeled as sterile. And so collectively, the results of these studies increase our understanding from the perspective of the human health regarding tattoo-related infectious disease. So I'd like now to switch gears and share with you some of the work that we have been conducting in my lab on alternative skin barrier models in the context of in vitro skin permeation testing. So an important criteria to evaluate the safety of a cosmetic ingredient is to know whether once it's applied to the skin, it remains there, or if it permeates the skin and actually has the potential to reach the systemic blood circulation, in which case it would have the potential to reach distal organ systems. So skin permeation can be evaluated either in vivo or in vitro. And for the in vitro testing, excised human skin is the current gold standard skin barrier. However, excised human skin is limited, and so it's difficult to procure it, and it's also costly. And so there has been an increased interest to identify reliable, non-animal, but human-relevant skin models that could supplement or potentially replace the use of human skin to predict thermal absorption of cosmetic ingredients and other products of interest to the FDA. So in our studies, we have been evaluating different commercial skin barrier models against the excised human skin using a very standardized experimental setup. And these alternative models include both reconstructed human epidermis, which consist of keratinocytes cultured at the air-liquid interface to differentiate into a structure that resembles that of the native human epidermis. And I'm showing here an example in the middle panel. And we are also testing synthetic membranes, which again are layered materials, this time non-biological, but that try to recapitulate some of the characteristics of the human skin barrier. And again, I'm showing here an example on the right. We're very happy in the lab to learn that our first publication in this area was accepted for publication. So actually it's online as of yesterday. And so I'm listing here our reference, and I welcome you to read it. And if you need a copy or if you'd like to provide some feedback, you're welcome to reach out to me. So in addition to these commercial models, we also partnered with the Tissue Bioprinting Laboratory at the National Center for Advancing Translational Sciences, or NCATS, which is a lab that is led by Dr. Mark Ferrer. And he gave an excellent talk yesterday, so I hope you had the chance to hear to it as well. And so his group has developed a bioprinted skin model, and we are evaluating the performance of this model for in vitro permeation testing as compared to excised human skin. And so this is a very active and close collaboration, and our modus operandi is that so NCATS manufactures the bioprinted skin, and then they ship to us overnight to NCTR, and then we conduct the permeation tests as well as evaluate barrier and histology. And so then we share the results of each lot of bioprinted skin with the group. We brainstorm on ways to improve the model, for example, by changing the cellular components, the media, the bioprinting protocol. And so our hope is that all this work will lead to the development of a suitable alternative human skin barrier model that meets FDA needs for in vitro permeation testing. So in addition, we have also recently looked into ways to capture the biological diversity of human population in terms of racial groups and skin pigmentation in our in vitro skin permeation tests. And the rationale for this work is that certain cosmetics are marketed to specific population groups, as well as some groups may disproportionately use certain cosmetic products, for instance, hair straighteners or skin whitening products. As well as that factors such as skin pigmentation may influence the absorption of certain chemicals. For instance, it's known that melanin can bind to cationic species, and thus it may have the potential to modulate absorption. However, studies in this area are quite limited and often contradictory, and so it is important for the FDA to develop methods that can test the influence of race, ethnicity, and skin pigmentation on the absorption of cosmetic ingredients and contaminants. And so we are currently conducting feasibility studies to meet this goal. So to this end, again, we are using excised human skin, but this time we are capturing the information about the race of the donors as well as pigmentation level. And for these, we are quantifying the levels of melanin using a colorimeter and spectrophotometer, as well as histological staining such as Fontana-Masson. And we are also, again, using reconstructed human epidermis, this time pigmented. And so besides the keratinocytes that I mentioned to you before, they also include melanocytes from donors of different races. And so these models can also be cultured in media with different levels of melanogenesis stimulating factors to induce a range of pigmentation levels in these models. And you can see here on the left bottom panel an example of these different pigmented models. And so again, we will use these skin models to compare the skin barrier and the absorption of cosmetic ingredients and contaminants of interest, and assess whether the differences among racial groups and skin pigmentation groups have any effects. Lastly, I just wanted to share a new initiative that we hope will inform on the dermal absorption of pair and polyfluoroalkyl substances, or PFAS. So PFAS are a large complex group of synthetic chemicals that have been found in some cosmetic products, both as ingredients or as impurities as a result of the manufacturing process. However, there is limited information on the safety and skin absorption of PFAS used in cosmetics. And so we are currently in the planning phase to conduct in vitro permeation tests using excised human skin. And of course, our hope is that our data will help OCAC make these determinations. And so I'll just like to finish by thanking everyone that contributed to this work, in particular, Dr. Kim and Kyuan from the Division of Microbiology, and Dr. Salman for providing material for this presentation. I'd like to thank the whole Office of Cosmetics and Colors group, which really has been a pleasure to work with, in particular Dr. Manga and Katz for their leadership and guidance, as well as Dr. Susie Fitzpatrick and all the collaborators on the skin model protocol, as well as funding from OCAC and the FDA chief scientist. And with that, I will finish my presentation. Thank you for your attention.
Thank you, Dr. Camacho, for that great presentation. Our last speaker is Dr. Patrick McDermott. Dr. McDermott is the Senior Science Advisor for the Antimicrobial Resistance at CVM. He is going to be speaking on the United States National Antimicrobial Monitoring System that we call as NAMS, that has helped ensure the efficacy of antibiotics. Dr. McDermott, the floor is yours, sir.
Okay, can you hear me okay, Raj?
Yes.
Okay, can you see the slides okay?
I can see the slide. If you can put that in the presentation mode, please.
All right.
Great, thank you.
All right, thank you very much for the introduction. Thank you for the opportunity to participate today. I'm happy to tell you a story about a FDA Center for Veterinary Medicine program and project to try to limit antimicrobial resistance in the food supply. And I'm speaking on behalf of myself, but also want to note Dr. Gregory Tyson and Dr. Heather Tate, who run the laboratory portion of the National Antimicrobial Resistance Monitoring System for the retail meat and animal pathogen section at CVM. So, you know, CVM's mission is to protect human and animal health. And when we're talking about antibiotic resistance, moving, let's say, from the farm to the fork, we're looking at a situation where human and animal health are at play and food safety and drug safety also are at play. So it really is sort of an intersection of FDA's responsibilities here in terms of protecting public health. We've been at it for a long time. We've taken seriously our obligation to try to limit the public health impact of antibiotic use on the farm. We know because we're using the same classes of antimicrobials in humans and food-producing animals and sometimes treating the same bacterial pathogens, there's certainly the opportunity for resistance to emerge on the farm, enter the meat supply, let's say, and then impact human health downstream. And so over the years, many things have been done at the policy and program level to try to assess this, and it's all aimed at antimicrobial stewardship in veterinary settings. How can we most judiciously use these drugs? Because we do need them to treat sick animals as well as sick humans. So can we minimize the impact of resistance evolving and preserve the effectiveness of these drugs for the future? So that's the general concept and the general policy we're trying to promote and better refine over the years. And this is an incomplete list of different things that FDA has done to try to grapple with this topic, but you know, it can be summarized as education and outreach activities, regulatory review processes that have a risk assessment built in to look at not just the safety and efficacy in the target animals, but safety in humans consuming products derived from those food animals. Greater veterinary oversight and the administration of antimicrobials, for example, going from over-the-counter to prescription status for antimicrobials has been a recent policy that's in development, still is in development, and getting feedback on, let's say. But there's also an active research area in this, and looking at the overt foodborne pathogens at different places around the agency, including NCTR and work going on in Dr. Foley's group. And then, of course, monitoring. You really have monitoring as the eyes of all of it, right? Do we know what the baseline levels of resistance are? Can we detect and do we have the sensitivity in our monitoring programs to detect the impact of different policies and hopefully the establishment of new lower baselines if the policies are effective? And then part of that monitoring is also antimicrobial use. And while we do get annual sales data by active ingredient, we don't have true on-farm use information like why antimicrobials were used, what was the route of administration, what was the clinical outcome. So we're working on that piece as well. So I'm going to focus. I really kind of want to tell a story more than show data, but I want to see if what could we say to the question about whether all of these investments and efforts have borne fruit. So again, our eyes are the NARMS program, the National Antimicrobial Resistance Monitoring System. It's been around for quite a long time. It started in 1996 with testing antibiotic susceptibility in human isolates of Salmonella in 14 states. It's now evolved over the years to include all 50 states for the human component. The other piece of it, the retail meat component, is conducted by the Food and Drug Administration, currently in 22 states and Puerto Rico. And we're looking at chicken, turkey, ground beef, and pork. And then in 2021, added seafood, shrimp, tilapia, and salmon. And we continue to do pilot surveys to look at other products raised with antibiotics and try to evaluate baseline resistance levels there as well. Our partners at USDA are looking at food animals presented for slaughter. They're looking at the corresponding food animals for the retail meats, which is chickens, turkeys, cattle, and swine. And also have recently done pilots look at veal, lamb, goat, and catfish at the processing plants. And we're all looking mainly at Salmonella and Campylobacter, but also E. coli and Enterococcus on the animal and food side as indicator organisms. More recently, starting in 2017-18, the Veterinary Laboratory Investigation Response Network, VetLearn at FDA, teamed up with USDA's NALM system, which is together a network of 45 diagnostic vet labs in the United States and Canada. And they're looking at animal pathogens. And this is part of our effort to more fully comport with the principles of One Health. We want to look at human and animal health. And so animal pathogens was added in 2017-2018. It's mostly from companion animals, but there are some food animal isolates in there as well. And they're looking at a number of organisms, including Salmonella, E. coli, and Enterococcus. After a science board review of the program in 2020, I believe it was, is that right? Well, fairly recently, let's say, we started looking and working more with the Environmental Protection Agency to look at the environmental component of the One Health paradigm. So as I'm sure all of you are familiar with the One Health definition, it's essentially a holistic approach that looks at human and animal health and the role of the environment in mediating the overall health. So healthy environment from the standpoint of antimicrobial resistance is something we're trying to help define. So we've been working with EPA to go out into the field, first by developing methods, which we completed last year. Those methods are now out in the field in some well-defined watersheds. And at the end of that, we hope to conduct a national survey in partnership with EPA's National Rivers and Streams Assessment to look at surface waters across the country and sort of get a snapshot of the resistance profiles there and how the organisms are compared to those we're finding in the other parts of NARM. So that's probably the most consequential and, let's say, novel direction we've taken recently in the program. And I list NIH here at the end because now we're doing whole genome sequencing on pretty much everything, most of our isolates, certainly all our Salmonella. And we deposit that data at the NIH, where NIH has developed tools to assign antimicrobial resistance genotypes to all the isolates that are there. And it's become now nearly a real-time program. The sequencing is done at the states and submitted to NCBI with as few avoidable delays as possible. So it's there for people to access. So we've got the system in place. We've got these policies we've put in place. Does the can NARMS help evaluate the policies? A good monitoring system ought to be able to do more than just give you baselines, but give you the impact of different, let's say, interventions. So a couple of examples here that I want to focus on for the rest of the slides here. So in 2005, CVM withdrew the approval of poultry fluoroquinolones, and based in part on NARMS data, but based mainly on a rising resistance rate in Campylobacter. At the time, we had no fluoroquinolone resistance in Salmonella, but it had gone up quite dramatically in Campylobacter. In 2012, another policy that was put in place was to put extra label restrictions on some of the cephalosporins being used on the farm. And this is another critically important drug class for treating human infections, along with fluoroquinolones. And then another policy development starting in January 2017, CVM no longer allowed the use of medically important antimicrobials for growth promotion. So medically important refers to drug classes used in humans and animals, and growth promotes an economic use of these drugs where they found many decades ago that antibiotics would increase the conversion of feed into animal weight. So it promoted animal weight gain. And it wasn't really a medical use, but mainly an economic one. And so that stopped, most countries have stopped this, and in January 2017 has stopped in the US as well. And then, so I want to see if what NARMS data show about these actions, and also overall, how are we doing as a country? We've been at NARMS since '96 and policies developing over the decades. It seems worthwhile for people to ask, well, how do we compare with other countries that have mature monitoring systems and policies in place? So I'll say a little bit about that as well. So in 2005, I mentioned the poultry fluoroquinolone approval was withdrawn, and I want to make sure I stay on time. This shows basically the status in the United States compared to many of the European Union countries. So if you look on the left-hand side, Campylobacter jejuni resistance rates here around 22%, about 3 times higher in 16 member states reporting in the European Union. So to the right of us with Australia, Sweden, Norway, and Iceland, those are countries that never approved them. So they still see resistance in their poultry farms for Campylobacter. But It's stayed pretty stable since it's ticked up a little bit, been fairly stable, and I would say plateaued in the U.S. I mentioned that Salmonella showed no resistance when that prohibition, let's say, was put in place. We've since seen it go up to around 9%, and I'll tell you more about why that is, and it's partly because of this international spread of serotypes. But again, looking at Salmonella, even though we're focused on Campylobacter, a knock-on benefit was resistance remains pretty low in Salmonella as well. On the right hand, you can just see the same data. That was data from humans. This shows data for broilers on fluoroquinolone resistance in Salmonella and Campi. Shows the average among EU member states and then the non-member states, which is mainly Norway and the UK, and the impact their data have on the overall picture. And then the United States on the right-hand bar. And it's important to look at this by serotype because resistance in Salmonella can be restricted by serotype. And so we can see, looking on the left, we're all concerned about Typhimurium enteritidis for sure. And the one that's appeared recently has been Infantis, which is spread around the world, and it carries decreased susceptibility to fluoroquinolones. And it appeared in 2014, some 9 years after the US stopped using those drugs in poultry production. So it's become a real problem. They have it in Europe. It's really become worldwide. It's not an unusual phenomenon in Salmonella to see this sort of global spread. And I think it's a vital part of the story because it does sort of point to the limited effect you can have if policies like this are only in nation by nation and not more international, through more international harmonization of these policies. In other words, it's a global One Health challenge that is hard to escape by yourself. Kentucky is another important serotype of fluoroquinolone resistance. They have a big problem with that in Europe. We've so far escaped that in the United States, but we have found this ciprofloxacin in multi-drug resistant Kentucky in travelers and also in food, imported food. So a big concern is whether that will gain a footing here like the Infantis has, which is spread, as I said, in the country, mainly in poultry, but now to some other food animal species. I may have to skip kind of quickly through this. It's hard to select the data. There's so much data that we might mention. I tried to highlight a few things, but in resistance in E. coli is a nice indicator because it doesn't have those serotype restrictions for resistance. It's a better general overall indicator of the selection pressure, let's say, and resistance in different sources. So this looks at resistance to critically important antimicrobials for broilers and turkeys. And you can see where the United States is on the right-hand side. It's sorted by ciprofloxacin resistance, but E. coli seems to suggest our policies to control or to be more refined in how these drugs are used has paid off, at least compared to other countries with the same sort of rigor in their systems and ways to detect differences. So it's not all good news. In the interest of full disclosure, we could do better on the cephalosporin side. Although we're still looking at pretty low numbers, the United States in human salmonella have about 3.4% resistance to extended spectrum cephalosporins. The EU average, I think, is 0.8. So there's an area where we could do better, and so we have attempted to do so with a policy to limit extra-label cephalosporin use in food-producing animals. The policy was announced in 2008, went into effect in 2012, and there's been quite a trend downward since 2009, certainly from about 36% in chicken meat down to around 12%, until our friend Salmonella Infantis arrived. It has not only decreased ciprofloxacin resistance, but it has the BLAS-CTXM65 gene, which gives cephalosporin resistance. And so we faced some headwinds when it showed up in 2014, and that's what the red lines indicate in this slide. The next intervention I mentioned was to eliminate economic uses, let's say, of antimicrobials, which started in January 2017. And as you can see here, the decline in overall annual sales of active ingredient went down 33% in the first year, but 38% from the high in 2015. And it remains right at the same level in 2021. This isn't a formal statistical analysis, but when we sort of take a cursory look at the indicator organisms from NARMS, and this shows poultry E. coli, you can see that the main classes that were affected by the production use policy generally trend down quite significantly, certainly since 2016 anyway, before the policy went into effect. And that's true for both turkey and broiler E. coli. And then if we look at a complete susceptibility, not resistant to any drugs, the United States is in pretty good shape compared to other countries we're collaborating with on these efforts. So that's a good indicator we can compare easily among different countries. In livestock, not as clear. Maybe there's some general trend down. At least the general trend isn't up for cattle sources. Swine pretty flat and even increasing resistance for ampicillin and gentamicin. So there are cases where it's not a simple correlation, even if we can't make causal inferences. I'm going to have to skip a little because I think I never have enough time. There's just too much data. This shows the pig data, not quite as good as the broiler data, but yeah, I just want to show how we compare on our indicator organisms across the board. Enterococcus, we survey or monitor for activity against gram-positive drugs or resistance against drugs with gram-positive activity. And again, it shows a pretty good, at least there's no major upward ticks except perhaps for QDA, which is quinupristin, dalfopristin. That was the only alternative for vancomycin-resistant enterococcus until a couple other drugs were developed later. So we still track it in NARMS. So that's an exception. So it's, a lot of data to choose from. I would invite you to take advantage of our interactive online tools to try to see how the story is evolving. It's present there in near real time now because we're putting up genomic data first and phenotypic data later. But there's a lot of wonderful tools. Maybe you heard Amy Merrill talk about some of them yesterday. You can look at the NARMS data. You can take information that links right back to NCBI and look at phylogenomic analysis of these strains. I mean, it's really quite elaborate, sophisticated tools, and I think user-friendly. And you can dig into these data more yourself as you have time, and I would encourage you to do that. But I think what we've learned so far from all this investment and all this monitoring seems to generally be positive in many cases. All right, it's always complicated because we're talking about different serotypes and sources, et cetera. But I think the policies have helped contain AMR, and that's what we're hoping for. We can't eliminate it until there are alternatives to antimicrobials, and even then, it could be difficult to reverse some of these trends. But I think the policies are demonstrably effective in helping contain AMR in many cases. And we continue to improve NARMS to get better data for better decision-making. The genomics has helped enormously in that. We have to keep in mind that multi-drug resistant salmonella and other pathogens, including other foodborne pathogens, do spread globally. There are several examples of it. And so national data can be skewed by strains that show up with pre-existing resistances that evolved elsewhere and spread among commodities and animals where those drugs are not used. And so, that's a vital part of this story. And so these strains that move can sort of undermine any nation's individual efforts to try to limit resistance. We don't completely understand all the drivers of resistance or the environment, and we continue to work on it. But I think overall, even though we need to be circumspect about the limits of what's achievable, I think what FDA has done in this area has borne fruit and warrants further vigilance going forward. And with that, I will thank you for your attention and look forward to the discussion. Thanks.
Thank you, Dr. McDermott, for all that you've done, especially the NARMS program for 25 years. So we will begin with our panel discussions. We have got some really good questions. What I would like to do is just kind of go through each of the speakers in the order they presented, and we'll have some open conversation after that. So my first question is to you, Dr. Liam. In one of your slides, you talked about the results on the usefulness of the ILMEREC organization on only to some extent. You highlighted the development and implementation of some harmonized strategies and identifying gaps on guidance and recommendations. Could you kind of expand on what are those harmonized methodologies and what kind of research or knowledge gaps that there was an interest in? Thank you.
Thank you so much for your question. I think it's multiple factors which are involved in let's say, the work where people are starting to work together a couple of years ago, which is the new alternative methodologies, and particularly in order to make use of them, let's say, in trying to speed up the risk assessments in particular. And this is an issue what also Dr. Steve Musser was indicating, listen, it is very important that we have still thousands of chemicals to risk assess. And it is quite important that we need to see how we can make use of modern technology, but also modern methodologies where you are making use of alternative approaches in order to have to combine the efforts of the different agencies working on this, and particularly there to see how we can let's say speed up the introduction and the implementation of these alternative methods in the chemical risk assessment. They can speed up the risk assessment, and certainly what we need to do together is to see which of the methodologies are competent enough in order to take on board in the chemical risk assessment. And there are lots of institutions which are dealing with this. Dr. Musser said also that there are a couple of partnerships working on that in the EU, which is very useful because we are combining efforts there. And I hope that it will also lead to a better uptake by the experts and by the users of the risk assessments, that we are getting convinced that these methodologies can be used and can speed up the time that we need for a risk assessment. It takes quite a lot of time to do a comprehensive one. And for that reason, we can make use of new methodologies to speed it up. That is.
Thank you, Dr. Liam. That's a very good answer. Just to dovetail on what you just said. So as a follow-up, what kind of opportunities do you see with working with the FDA when you're addressing these risk assessment related to either the new alternative methodologies or chemical toxicants? Because as you're aware, different countries have different priorities and different regulatory decision-making bodies. So how do you see working with the FDA as it benefits the global community?
I think that what we were also already were given attention to is the issue of validation and qualification of new methodologies is an issue. It's taking a lot of time before you can validate methodologies. And certainly if you look into what kind of aspects should be considered in the validation, we really need to see how we can do that in a more efficient way, particularly the regulatory uptake. In order to have the risk assessment agencies doing this, they are really in need of working together in order to see how we can do that in the best possible way. And of course, we need to have approaches then taken on board, which is for which we need, let's say, to join efforts, because I really think EFSA is a small agency compared to many others, and I think that we need to work together on that. So that's the only thing that we would like to continue doing so. We have already started a couple of years ago with colleagues of the US FDA, and we do that also together with EPA and other agencies. It's very important that we continue with this.
Thank you, Dr. Liam. I think I agree that collaboration, that partnership between our countries and our organization is the key in driving these public health missions. I would kind of turn over to Dr. Musser. Dr. Musser, and this is also to Dr. Liam. One of the questions that was asked in our Q&A box is, could you comment on the current state of NAMs that have been developed to model population-level variability and individual susceptibility to chemicals? And also, what are your thoughts on using primary human cell lines from diverse human population? Thank you.
It's a great question, and I didn't answer it online because I wanted to do it verbally since it'll take some time. I think this is one of the biggest promises for NAMs in general, is the ability to look at sensitive populations or underrepresented populations. I mean, a lot of times when we try to do these evaluations, we're looking at a fairly defined area or group that may not represent the human population. And I think this area still has a long way to go, but in this particular case, looking at different population level variability is an incredible promise for NAMs in general. And I certainly encourage all the work that's going on in here. I think, access to the cells can be problematic depending on where they come from. There's ethical issues sometimes with whether they come from where the cells actually originate from, and they'll have to be overcome. And there's other ways, of course, of developing them that can get around those concerns. But all in all, I just see this as being one of the most important aspects of NAMs, should we be able to get them off the ground.
Thank you, Dr. Musser. Dr. Liam, did you have anything to add to that?
No, not really. I think it was well captured. I think that's the challenge that we have in developing the development and implementation of the NAMs is particularly to make use of what we have learned from let's say, all the developments, and particularly from validation experiments, in order to also see what is the battery of the different types of NAMs which have been, let's say, taken into consideration so far. If there would be a battery, let's say, particularly for better predicting the toxicology of particular chemicals. And we are not yet there. And indeed, it will take some time before we have really established, let's say, these strategies in order to speed it up and to get also confidence in the outcomes.
Thank you, Jean. Dr. Camacho, you have raised your hand.
Yes, I just wanted to share some thoughts on this, the importance of trying to capture the diverse population using these NAMs. And I think that's a very important question. What is the sample size of the assays, right? So if we use, say, rats, we use each individual animal as one individual, as one N. When you're doing NAMs, so based on our own experience using this reconstructed human skin, it was right in, already along the way of the work that we realized that even though we're purchasing different batches from this company, all the batches were manufactured with the same donor. So meaning that we're in essence doing replicates, technical replicates, rather than biological replicates as we had thought. And so sometimes you really have to ask the questions and understand your system, especially if you are not doing it in-house. So if you are using a commercial source of the model, because you may be working with something that is not as representative, as not as biologically diverse as you may think. And then the other thing is, as I mentioned, so in our work we are also trying to capture skin pigmentation and race, ethnicity, diversity in our models. And so there's ways that the models can be made more complex to really try to include different cell types and different represent different human population. But asking the right questions and making sure you understand the biological variability in your system, I think it's critical.
Thank you, Dr. Camacho. The next question I have is for Dr. Foley. So what are some of the new approaches that are being developed to understand the pathogenicity of Salmonella? You mentioned that in your concluding slide, and maybe Pat, you can kind of jump in as well, since you talked about Salmonella. So Dr. Foley, please go ahead.
Sure. I think kind of piggybacking on the NAMs discussion as well, too. I think that's an area where there's a lot of growth potential in there, trying to get model systems that are non-animal model systems are, I think, to look at different stages in Salmonella pathogenicity, trying to understand how these models look and match up to the real kind of the in vivo system, if you will. I think that's an area there, whether that's using three-dimensional cell culture or those different cell lines, that kind of stuff, to more effectively model different things, because a lot of the traditional work's been done with two-dimensional cell culture, and data kind of shows that provides data, but it may not be the same as 3D culture, time, space tend to make a big difference in a lot of the ways that the bacteria interact with the cells. The role of the microbiome as well too with these as well, trying to understand how these interactions in the gut or other areas play a role as well too, because they're not. A lot of our stuff's been done in monoculture kinds of stuff, but they're not. But when you're taking them into the intestinal tract, it's not a monoculture that you're dealing with, and so trying to understand those interactions. If one bacterium does some takes in nutrients, that kind of stuff. There's the kind of the byproducts, that kind of stuff. Salmonella may be able to use those, or other organisms may be able to use those for different things. Trying to understand that the cell-to-cell communication is related to that is key as well. And I think developing improved genetic tools as well too, to knock out genes specifically, and then have those in vitro systems to assess the function when you do knock those out. And one of the challenges with our plasmid work, for example, is plasmids are often in a cell in multiple copies. And so you may go in and knock out the gene in one copy of the plasmid, but the other, if you haven't knocked it out, now you may have kind of borrowing from the eukaryotic genetic front, you may have like a heterozygous kind of genotype in there where it's knocked out in one, but present in another plasmid. And so trying to get at tools. And so that's one of the things that we've been trying to do, is develop tools to be able to knock it out in all copies of plasmids and then assess that. And there are lots of good tools for the genome, the chromosome, not so much for plasmids. And so that's an area where I think there's improved opportunities.
Thank you, Steve. I think you answered one of the questions on the Q&A box where they were asking about the human safety assessment of human cell lines from the NAM's perspective. Thank you. Pat, did you have anything to add to what Steve said?
Well, I would say in terms of alternative methods for improving FDA's work, CVM's work, I'd say, in the AMR and the antimicrobial resistance field, I sort of think of the question in two ways. One is, what we used to do is we get all these isolates sent to a central laboratory, grow up the bugs, we plate them on 96 well plates with two-fold dilutions of antimicrobials, measure the minimum inhibitory concentration, send those isolates to another lab, have them chew them up with enzymes and do pulse field gel electrophoresis to compare strains. And then if we had time and resources, we might do multiplex PCR to look for what genes are there. And all that's been supplanted now by genomics. So in a sense, we have the level of resolution and timeliness for monitoring that we need. It looks more and more like the genotype could predict the phenotype, and perhaps the MIC with a great deal of reliability. So we're really in the phase of just trying to fully exploit this technology. And what that means for us is better machine learning tools and automated processes and things of that nature. Now, if you go to the other side of the issue, what are the alternative methods needed to grapple with AMR? I would start with alternatives to begin with. I mean, part of this is a business problem as well, and trying to, companies are pulling out of the field and developing antimicrobials because, well, why would you want to develop something that you're encouraged not to use, in a sense? So we need alternatives. We certainly need alternatives in the animal field. And, people are working on things like phage and immune augmentation and things like that that could obviate the need for these medically important antimicrobials and perhaps intercept the chain from farm to fork that compromises human health. So I think on the monitoring side, well, I should add to that, maybe one more thing, and Steve mentioned this a little bit, is the metagenomics approach. That's another area that needs more complete exploration and exploitation. It does get you beyond a lot of limitations of growing bacteria, and so you can get a big picture snapshot of the resistome from any complex sample. And while it's got problems of reproducibility in terms of being a surveillance tool, we're working on even that aspect with pre-enrichment steps, that might make it more reliable and controlled, so sample to sample. So I guess I'd leave it at that and answer it in those two ways.
Thank you, Pat. And I think yesterday, Amy Morrell and Dr. Shu gave a really good presentation on how to use the genomic data and machine learning capability in antimicrobial resistance. Thank you. I'd like to move to Dr. Camacho. I have two questions for you. One question was, and maybe Steve, if you want, Dr. Musser, if you want to chime in, will NAMs, Luisa, impact testing of PFAS?
I'm not sure I understand the question. So, but okay, PFAS in the context of cosmetics, which is what we are, foods for Steve, which is what we are dealing with today. So OCAC, they have been, so the Office of Cosmetics and Colors, they've been following, the use of PFAS added as ingredients through some years now. And so because, of course, cosmetics are applied to the skin and there's not much information about the dermal absorption of PFAS, so that's the interest of our work. So in our case, we are using, we are going to start at least with the in vitro skin permeation testing using human skin, but we could also try to use the reconstructed human skin. Now, those models are not yet ready for prime time. Their barrier function is much weaker than skin, the excised human skin, but they do have the advantage that it's a very controlled experiment. So these models, so they'll be considered NAMs because they are 3D models in which we apply in this case, the PFAS to the top of the skin. That would be not a challenge to those. For NAMs where the cells are in a media, there may be some challenges there with the dosing of the cells because of the solubility of PFAS. So there's, kind of challenges using NAMs and PFAS. Just to add to this, the other challenge we are having with PFAS is also the analytical methods. So, they are not available for all PFAS of interest. And so that's, part of the discussions we have now is how to address that and be able to provide OCAC with the data they need. So I'm not sure I answered the question. Maybe Steve, well, Steve and Steve may add to this.
No, I think you hit it pretty much on the head there. The problem with a lot of these tests, and PFAS is kind of a perfect example in general. I mean, it's a very broad class of chemical compounds, hundreds of compounds, maybe more, is that they're very insoluble in cell media. And, if you're dosing them in zebrafish or cell culture or something like that, you don't know if it's sticking to the plastic or sticking to the glass or sticking to any of the other things. And so if you get a negative answer, you don't know if that was because the cells weren't exposed or the system wasn't exposed. And if you get a positive example, you don't even know, well, was it a lot of it? Was it most of it bioavailable? Was it some other problem? So I think PFAS point to one of those real challenges in using these tests is in, how does dosing and exposure really fit into some of these NAMs and evaluating NAMs? It's just one more of the questions that we need to know when we look at using NAMs in regulatory context.
Thank you, Steve. One quick technical question, Lisa. How do you purify and quantify melanin concentration from human skin?
So to quantify, so we do not isolate per se the melanin. So what we do is we digest the skin and then we just transfer the solution to a spectrophotometer, and then we quantify based on the absorption spectrum of melanin, which is known. So that's one method. The other method we do for skin pigmentation is just use a colorimeter. And so, again, based on luminescence and other parameters of the skin, we can relate that to skin pigmentation levels. And the third method that we are using, just to kind of be comprehensive about, trying to categorize these different skin models into different groups of pigmentation levels, we are also doing histology and staining that specifically stains melanin. And so in this case, the chemicals, the staining is called the Fontana-Masson, and there are several references in the literature with these methodologies.
Thank you. Great answer. On a lighter note, I'm thinking about getting a tattoo this weekend. What do you think?
The what, sorry?
I was thinking about getting a tattoo this weekend. Should I get it?
Okay, so actually, FDA and OCAC has several very helpful websites for consumers. One of them is Think Before You Ink, which I think is a great punchline. And so I'll welcome you to read those because I think you'll find a lot of useful information there from the consumer's perspective. From the manufacturer's perspective, actually, I just learned from Dr. Katz yesterday that OCAC just published or posted the draft guidance for manufacturers and the distributors of the tattoo ink products. And this, maybe Steve can better talk to these, but my understanding is that it's to provide some guidance, which is now open for public comment on how to identify situations where the ink products, tattoo ink products may be contaminated or how to avoid these situations. And so there's a lot of useful information for consumers, manufacturers in the OCAC website, and I'll welcome everyone to have a look.
Thank you for that. I'll have to think twice now to get that tattoo. My last question is to Pat. Pat, what are your perspectives on the current challenges in the antimicrobial resistance sphere, especially in food-producing animals?
Well, I think there's probably a lot of people would say the first answer to that is the increasing need for protein, right? And the need to produce more animals as the global population works towards its peak. So there's more demand than ever for food animals as a source of protein. That means raising a lot of them. That means an expectation that more use would be required. So more than ever, we should be working together on this, I think, internationally and trying to push the development of alternatives, promote the policies that optimize judicious use. I think, and find ways, I mean, there's a lot of elements to the solution, but find ways to incentivize industry to invest more in it as well. There's no simple way out. It's just going to take sort of hard-nosed science again to look at alternatives and new products and dedication to a global policy that promotes judicious use. Because like I said, you can do it all right in your neighborhood, but if something blows over the hedge, then you might be back to square one. And that's a good sign that it's not going to be possible with piecemeal attempts in different locales to try to do this. I think it needs a more cooperative approach, and that can be very complicated. So yeah, it's a big challenge, Raj. It's a big challenge.
Thank you, Pat. Steve, do you have anything to add real quick? Steve Foley.
I think
Sorry. You have two Steves.
I think what Pat said, I agree with what he said. I think the challenge is going to be the international piece of it and trying to, like you said, there's a lot of things that blow over the hedges that can create problems that, are outside our control initially.
Thanks, Steve. With that, I think we are very close to the end of this session. I'll give a last chance. Does anyone have any? We have 3 minutes. Any last minute comments?
No.
Okay. So I just want to thank all the speakers for their time and for sharing their scientific perspective on such diverse topics, and also to our staff who have been working tirelessly behind the scenes. Thank you all, and enjoy the rest of the Science Forum.
Thank you.
Thank you very much.
Bye-bye.
Bye-bye.
So thank you all so much for tuning in to part 1 of day 2. We are going to head to lunch for about an hour. We will return for our next session at 12 p.m. Thank you, and we will see you soon.
All right. I think we need to start. Good afternoon and welcome to FDA Science
Recording
Forum.
in progress.
All right. We need to start. Good afternoon and welcome to FDA Science Forum Session 7. My name is Manju Manjanatha and I'm from the National Center for Toxicological Research. And I'm moderating this session with Julie Snyder from OSC. As you're aware, this session highlights advancing products based on novel technologies. We have 6 excellent speakers for this session. The first speaker is from outside of FDA, and the rest are within the FDA from different product centers. The external speaker gets 30 minutes to speak, and all the internal FDA speakers get 15 minutes each to talk. Speakers, please stay back until the end as we have a Q&A panel discussion for 15 minutes. Attendees, please type in your question or comment into Q&A box for me to read it out to our speakers. Well, with that, I would like to start by calling our first speaker, Dr. Catherine Wu. She's from Dana-Farber Cancer Institute and Harvard Medical School. And her talk this afternoon is on update on personalized cancer vaccines. Dr. Wu, take it over.
Thanks so much for that kind introduction. I will start to share my slides. Just give me 1 second. There we go. Okay. Can everyone see that?
Yes, we can.
Yeah. And is it in slideshow? Let's see.
Yes, it's in presentation mode.
Okay. How's this?
Looks good, and we hear you.
Okay. Terrific. Great. So thank you so much for that warm welcome. And I feel very privileged to get a little bit of extra time to speak. So thank you very much. I'll just start off real quick by just saying that, I think we do understand that our world has really been rocked and changed over the last couple of years. I think that our cognizance of vaccines and what they can or cannot do for us is more than ever through our whole pandemic effort. I want to remind you that when we talk about the SARS-CoV-2 vaccine, we're really talking about prophylactic vaccines. And for the most part, we've been talking about antibody generation, but with some consideration to T-cell responses. What my work has focused on is really cancer vaccines. And here, this is really therapeutic vaccines for the most part. Of course, we hope in the future that we can get to prophylactic vaccines. But for now, by and large, we're dealing with patients who already have cancer. We're using a vaccine as a therapy with the goal that under the appropriate antigen stimulation, that we increase the breadth and diversity of a tumor-specific T-cell response. I like to think that a lot of the work that has been going on over time in cancer vaccines has actually helped to fuel the rapidity for which we saw the COVID vaccines roll out. But I think we're also at a time now that we're in this completely different era, that we can learn lessons from the pandemic that will in turn help us generate better cancer vaccines. So I'll talk a little bit more about that later. Let's see. Here we go. So I just want to remind everyone, in terms of the immune system, our immune system is hardwired to interface with our environment and to deal with heterogeneity in the external pathogens that may come in different shapes, sizes, colors. But I also, at the same time, want to point out that interfacing with infectious pathogens and cancer are not the same. We know this, but I think just from immunologic perspective, infectious disease provides us with the advantages of foreignness and high immunogenicity. In cancer, we're dealing with a lot of genomic heterogeneity. We're also dealing with the capacity for cancers to immune evade. And so that kind of sets up the situation where we have to work a little bit harder. And from the immune perspective in cancer, we're dealing with kind of a scenario of chronic inflammation and antigen exposure that can lead to T cell exhaustion. And so this is another way of kind of depicting this kind of challenge for us, which is that in terms of our cancers that we're dealing with, these are fueled by the heterogeneity to generate clonal evolution, and so unfortunately, typically by the time we see cancers, they have already evolved to a point where they can immune evade. So again, we have a parallel host immune system, but the cancer can either intrinsically evolve to lose tumor antigens or can see the expansion of mutations that affect antigen presentation or immunoregulation that can either directly or indirectly spur on the presence of inhibitory cells that dampen the impact of our immune response. And so I think when we think about from a therapeutic standpoint and trying to harness the various different capacities of T cells, and there are many different capacities of the T cell, we really see in terms of immunotherapy, really different types of immunotherapy that have kind of been springing up in our landscape, each that are trying to target or really exploit particular aspects of immunity. So for the purpose of this talk, I'm really going to focus on vaccines, and I apologize for the poor quality of this reproduction, but for this talk, we're going to focus on vaccines. I want to just have you focus on two colors, the pink cells versus the green cells. The pink cells are those pre-existing T cells. So some therapies do build on pre-existing T cell immunity, so TIL therapy and also checkpoint blockade, whereas vaccines, other therapies such as our T cell gene therapy, our CAR T cells, these are synthetic sorts of gene therapy where we're kind of sending in T cells in numbers that we didn't have before. I think vaccines have the unique aspect where they can both amplify not only pre-existing T cells in the pink, but also elicit novel responses, and that's in the green. And so I think that that's a very unique and interesting aspect of vaccines that have important clinical implications. Let's see. Okay. And I think the other challenge that we have to think about, again, thinking about the tumor immune interface, is that we recognize that not all tumors actually allow the immune cells to come in and occupy the same microenvironmental niche. So by now, we know that some tumors in the middle panel can be inflamed. Other tumors tend to exclude T cells at their rim. And then there are other tumor cells that may even generate these tertiary lymphoid structures that appear to be beneficial. So I think there's really quite a variety of different sort of configurations that immune cells have in relationship to tumor cells. But I think in terms of the vaccine, conceptually, what we have been thinking about over time is that this is a very unique sort of therapeutic modality that allows us to steer the immune response. So in other words, if we do have a therapeutic modalities that can help release the brakes, like the immune checkpoint blockade antibodies, and we have good adjuvants that can kind of accelerate the response, then can we not use the vaccines as a modality to steer the response in the direction that we want, namely in a highly specific anti-tumor immunity fashion, and yet with fewer side effects on vital tissue, so minimizing toxicity. Now, if we're going to go in the direction of vaccines, what are we going to use to immunize? And this is kind of my bridge to talk about neoantigens. And neoantigens are those antigens that can be immunostimulatory that are arising from mutations. So, and they are, they're not immunogenic in the wild type sense, but then with the mutation, there can be an alteration. At least some of the mutations will generate alterations that can get transcribed and translated as altered amino acids, which in turn then at least a portion of them can bind to HLA and find themselves being displayed on the surface, where those peptides can be recognized by T cells. And the concept of neoantigens is not a new one. For decades, it's been recognized that tumor neoantigens would be truly the optimal tumor antigen, because by virtue of being born of mutation, they are exquisitely tumor specific, and at the same time akin to pathogens, because they are somatic events. There is a lack of central tolerance on the part of T cells for targeting them. So they're highly immunogenic. And this is really quite different than where we've been over the past few decades, thinking about targeting shared overexpressed tumor self-antigens that are variable in the degree that they are tumor specific and variable in the degree to which they are immunogenic. What has changed for us, and I think this is why this is in the novel technology section, is that over the past 10 years, we've seen really a technological transformation with the availability of next generation sequencing that has allowed us to readily identify those tumor neoantigens on an individual cancer by individual cancer basis so that they can be targeted. And so by now, many groups, including our own, have generated workflows that look like this, where we have pipelines that take DNA from tumor versus normal. We can perform whole exome sequencing. How did that happen? Can we go back? Okay. With HLA typing, we can also use predictive algorithms to focus on those alterations that are expressed. So we have RNA sequencing coming in as well, and then to predict what are those HLA binding peptides related to that mutation that has the potential to bind to that personal HLA. And if we can do so, then we are set with finding candidate neoantigens that in turn can be targeted, for example, in a vaccine. And this type of kind of analytic approach has led us to consider that this is a paradigm shift in terms of what we can do therapeutically, that we can target a tumor not against one determinant, but multiple. So getting to that kind of question of multi-pronged attack and dealing in a multi-polyclonal fashion. So addressing head-on the challenge of tumor heterogeneity, but also not targeting normal tissue. So getting us away from toxicity. And by now, when sequencing first came out about 10 years ago, we tried to conceptualize how this could look in a therapeutic arena. So, could we not in real time collect tumor tissue and a source of normal tissue for germline DNA, perform exome and RNA sequencing to identify specific mutations, as well as HLA typing that would allow us to predict those personalized HLA binding peptides, and then come up with a manufacture strategy so that we could create those mutated peptides for targeting. The approach that we chose was long peptides, because of the wealth of safety data that preceded our work that convinced us that this would be a very safe approach in our patients. At that time, there was very little, if no information on mRNA as a vaccine therapeutic. And we also wanted to go with a long peptide, so 20-30 mers, that by prior published data had demonstrated that this would require a professional antigen presenting cell to internalize the long peptide that would be able to harness through processing and presentation both the CD4s and CD8 cells. And this we put also together with poly-IC-LC, and our hope was also to bring this together with a checkpoint blockade inhibitor. And so I don't want to go through a lot of this because this is published data. Our studies are here in the red, but we and others demonstrated in the adjuvant setting that for both high-risk melanoma and newly diagnosed GBM, that this was safe, feasible, highly immunogenic, and in fact, across the studies, promising in combination with checkpoint blockade. And just to give you a little bit of flavor of what we observed, these were 2 of our initial patients that we treated who had stage 4 disease, melanoma adjuvant, and not surprisingly, after their vaccine, they did have a recurrence of their disease. And then their treating physician started them on anti-PD-1 therapy. But what was surprising was that both patients then had complete clinical response that appeared to be enduring. We were able to document that there was actually strong neopeptide specific responses in the red across a titration of different peptide concentrations, but largely not against the black, the wild type peptide. And on radiograph, you can see that after PD-1 was initiated, within 12 weeks, we could see regression of those lesions that had happened, that had been detected at the time of recurrence. So our initial work was really focused on trying to monitor responses immediately after the priming and the boosting, but over time we have had the opportunity in our academic investigator-initiated trial to follow these patients over time. And we had the opportunity to look at these patients 3 to 4 years later. In the dark gray was the time intervening from the first publication. And all of our patients generously provided additional peripheral blood to us so that we could study these responses. And by and large, we were able to detect very promising short and long-term immune activity. Just going from left to right, we were able to look to see the effects of the vaccine on the transcriptional signature of neoantigen-specific T cells. What we did was we generated tetramers that allowed us to directly pull out these T cells from the peripheral blood, and then we performed single-cell RNA sequencing. And I think you can appreciate across the different patients that we defined these 4 different signatures. And when we started to look in a time-resolved fashion, we could in fact see that there was kind of a naive signature going to cytotoxic immune contraction and ultimately memory generation. So this was great news because this is certainly what we hope to see with a vaccine. Using that same data, looking at T cell clonality, looking at the T cell receptors, we were able to identify T cell diversification and persistence. So in these pie charts, you can see each color denotes the first time that we detect over a time course a clonotype. And what I hope you can appreciate is that the red that we detected from the very beginning does persist over time, but we see the addition of more colors as we shift from the left to the right. So again, speaking of the diversification, and even more so when we did a functional assay such as the ELISpot, which detects interferon gamma secretion, we could see even close to 4 years later that there were still very strong circulating immune responses to the same immunogens that we had used in our original vaccine that we could detect at week 16 after the first boost. Because we had single cell TCR sequencing data with the alpha beta chain information that allowed us to definitively define a T cell clone, and we knew which of those were neoantigen specific, we could see that neoantigen-specific T cells were tracking to the site of tumor. And just to lead you through this, this is one of the patients that I talked about before that had relapsed after the neoantigen vaccine that then got anti-PD-1 therapy. And based on peripheral blood, as I mentioned before, the overlapping green and orange circles tell us that there's a large overlap in terms of the T cell clones going across time. So this persistence of neoantigen-specific T cells. But what was interesting to us is that by using that molecular information afforded by the TCR single-cell sequencing, this gave us molecular barcodes to then evaluate the bulk TCR sequencing. And we could find that in the recurrent tumor, that there was molecular evidence of T cells coming from neoantigen specificity that were tracking to the site of tumor. And so one can speculate that perhaps the reason why these patients responded so well to the anti-PD-1 is that the vaccine was able to actually generate T cells that were able to track to the site of the tumor and that were at the ready for anti-PD-1 impact. And finally, we could look and see that there was evidence of epitope spreading. So in this adjuvant setting, epitope spreading can serve as an approach that allows one to look for evidence of on-tumor targeting, because it's evidence that there's a secondary wave of immunity arising from tumor kill. And so in our group, we were able to detect epitope spreading even after vaccine alone, persisting even at the time of anti-PD-1 therapy and beyond. In other patients, we could still see evidence of epitope spreading 3 or 4, those expanded epitope reactivity even 3 or 4 years later. And an independent group also looked at epitope spreading in the setting of neoantigen targeting vaccines and could see that this was associated with improved outcome. So I think, in terms of where we are across different neoantigen targeting therapy studies, I talked about our studies. There are other groups that have obviously been working on this as well, all with very promising immunologic results. I already talked about the complete responses that we saw after anti-PD-1 therapy with our peptide-based vaccine in melanoma. Ugur Sahin's group using RNA vaccine approach in melanoma saw decreased number of recurrences. The group Neon Therapeutics evaluated epitope spreading, saw this in melanoma, non-small cell lung cancer, and bladder cancer. Vinod Balachandran at Memorial Sloan Kettering, and now this paper has since been published in Nature, has demonstrated the lack of any recurrences after an RNA vaccine in pancreatic cancer. I've talked about the tracking of T cells to the site of tumor. We and many others have seen the same thing. And more recently at the AACR meeting, there was the first randomized open label phase 2 trial that was reported out by Merck Moderna for resected high-risk melanoma that seemed to show a promising clinical outcome. So going forward, I think our general approach has been to continue to do very intensive bedside to bench and back again sort of efforts. And I would characterize very briefly, for the sake of this talk, 3 general areas that we've focused on. So one is by virtue of the fact that we're performing our vaccine studies in different disease areas and the wealth of kind of rich tissue material that we are collecting from our study subjects. This does give us the opportunity to perform disease-specific research. We are very interested in ongoing clinical trials that look across different settings, because each disease setting does afford us new and different opportunities to learn. And finally, we're always thinking about new capabilities that kind of enhance our ability to understand immunologically what our interventions are doing. And so I have a slide for each of these 3 different areas. So first, in terms of disease-specific research, I think, one area that we're very focused on right now is trying to learn more about the phenotype and character of tumor-specific T cells at the site of tumor at baseline and the impact of therapy. And in our initial studies, we've used single-cell transcriptome sequencing to try to understand the phenotype, link this together with TCR information. And because of the general rarity of TCRs with anti-tumor specificity in the immune microenvironment, there being bystander T cells that lack anti-tumor function, we've found it imperative to try to take that TCR information and link it and try to understand the specificity of those T cells. And in terms of antigen, this has meant either looking at reactivity against tumor or reactivity against different epitopes, whether they're coming from the mass spec or from prediction or from various established peptide pools. And in one exciting study that instructor junior faculty member in my group, Giacomo Oliveira, has performed is he's looked in the melanoma setting, and he really came up with an ingenious way of cloning TCRs at high throughput, using that CDR3 alpha beta chain information so that he could kind of in parallel clone hundreds of TCRs together. He also came up with a way to do combinatorial dye labeling of each of the clonotype bearing cells so that we could with a simple flow-based assay, be able to discern patterns of reactivity. And as I mentioned before, that these TCR-bearing cells could be tested against tumor, for example, or specific antigen targets. And shown here is just an example of the readout using 41BB, CD137 as a readout by flow cytometry. You can see in this example of this TCR that there is reactivity against autologous melanoma or interferon gamma secreting melanoma, but not against autologous B cells or PBMC. And so this work has since been reported, so I'll just summarize briefly. Lots of pearls that we've kind of gained looking deeply at the TIL landscape of melanoma, which I really think is this TIL analysis really becomes some sort of tumor immune cell archaeology where we're able to see past hallmarks of kind of immuno editing going on. And it seems to be a graveyard of T cells that have really potent tumor specific recognition, but they are exhausted, they are tired. Those specificities may be directed against melanoma associated antigens or neoantigens, and it really seems to vary from patient tumor to patient tumor. These appear to be polyclonal responses and quite heterogeneous. On the CD4 side, we see very many similar aspects, and again, I won't go through it here, but we also see evidence of direct engagement of immunosuppressive CD4 Tregs as a mechanism of immune evasion. And so I think one of the takeaways is that the T cells we want the most are exhausted. Can they be revived or replaced? And I think this is, again, a place where vaccines, knowing that this is the baseline, vaccines may really serve to overcome this phenotype. And this is something that we're really quite interested in. Our ongoing phase 1 clinical trial efforts, you can see here, these are different Dana-Farber clinical investigators that I've really had the privilege of working with. And we currently have 6 or 7 different clinical trials ongoing, each testing different aspects of vaccinology and different stages of disease that we hope we'll learn. Some of these stories are coming to fruition, and so we hope to share it with you sometime in the future. And finally, in terms of looking at our our toolbox of immune monitoring tools, where we've become quite interested in spatial transcriptomics, and I've had the privilege of working closely with Fei Chen at the Broad, who has been the pioneer of the slide-seq technology, which is really an approach to take tissue and to fix it onto beads that have X and Y coordinate information encoded in them so that they can go through somewhat like a single cell transcriptome like protocol, but then there is the ability to computationally reconstruct the information later. I think where our group has come in is building on Fei's RNA platform. We've been able to take our targeted TCR-based approach and link that together. So now we can not only look at the phenotype of those T cells in a spatial distribution, but also T cell clonality. And in one renal cell carcinoma pulmonary met, where there was an interface between normal and tumor tissue, we could look to see which of those clones were tumor enriched versus tumor depleted. And we have many, many interesting studies ongoing using this technology. I'm going to skip this. But only to say, I think the field of vaccines has really been quite a roller coaster over the years. There's been the ups and downs, but I think that we're in a very interesting moment where there's been a lot of foundational different studies. But I do think that, and I think neoantigen specific T cells are undeniably critical to the immune control of tumors, and therefore for most forms of immunotherapy. I think vaccines that can induce those T cells are expected to be a powerful synergistic partner for most forms of immunotherapy. My colleague Ed Fritsch pulled together this kind of graphic some time ago just to think about what are areas that we really need to focus our attention. And there are many in order to kind of, quote unquote, get there. Neoantigen sources are one, because right now we're really limited to just thinking about somatic mutations that are either single nucleotide variants or insertion deletions, but we know that there are many other different types of alterations that are out there that might be more prevalent in certain types of malignancies that we might say are otherwise low tumor burden. And we also, in order to kind of hit the big time, we have to kind of address the issues of cost and time. And I think we got to get there a little bit faster. And this will really impact the kind of scale and the indications that tumor vaccines can be used for. So just in terms of some final parting thoughts, I think that personal antigen targeting does address the fundamental challenge of cancer heterogeneity. There have been encouraging results, and we've been very appreciative of our strong cooperation with motivated patients that have enabled such studies to be undertaken. I think the question that is coming up more and more, which is how do we approach true prevention? What is the right setting? I think I can very much envision how we could focus on looking at early disease, early cancers, or following consolidated therapy. And I think, again, how do we address the challenges of cost and time? And have we learned from the pandemic that can help us get to scale? So these are our challenges coming up. And I want to thank you for your attention. I'm really beholden to my fabulous group who has focused not only on the tumor aspects, but also how we can harness T cells to eradicate tumors. And these are my funding sources, and I thank you for your attention.
Thank you, Dr. Catherine Wu. That was such an impressive talk. And also thanks for sharing that data, very impressive, and the work you've been doing on cancer vaccine and the update that you gave us. Well, I hope attendees have questions. They can enter it into Q&A, and then we can come back and ask Dr. Wu. All right, let's move on to the next speaker. He's going to speak a slightly different topic, and he's Dr. Wenming Shao. I hope I didn't butcher his name. He's from Cedar, and he's going to talk to us on next generation sequencing technologies in B-cell receptor-based immunome profiling and minimal residual disease biomarker discovery. All right, Dr. Shao, take it away.
Great. Thank you for the introductions. Can you see my slides and hear me well?
Yes, we do.
Okay, great. Again, my name is Wenming Xiao. I'm a lead bioinformatics scientist in Office of Oncological Disease. And I'm very grateful for the opportunity to present you our study on evaluation of the PCR sequence profiling with NGS technologies. So this is the ongoing study, and therefore I would like to take this opportunity to collect feedbacks or suggestions from the audience and adjust our study design if necessary. Okay, let me start with a summary report published by our office early this year. In this report is looking into all NDA or BA submissions for the treatment of the immunological malignancies between 2014 and 2021. And look at the use of MRD data to support drug approval. Most of these submissions were using PCR or TCR sequence as a MRD biomarker. Of 41 applications with MRD supporting data, nearly 40% of them could not be included in US prescribing informations, mainly due to analytical issues or assay performance issues. The conclusion from this paper is that there's an urgent need for the improvement of assay validations, standardizations, as well as the sample processing and sequencing. Meanwhile, the community is still evolving and improving the technology for PCR sequencing, as well as the usage of the PCR sequencing in the disease patient, as well as in normal patients, normal peoples. And there are lots of the bioinformatics tools being developed for this kind of applications. So with that, we started this project in focus on the PCR sequencing technology with three major objectives. The first objective is to fill the knowledge gap of PCR-seq in terms of assay, sequencing technologies, analytical tools, establish the QC metrics for reliable result, and determine the assay detections, sensitivity, accuracy, as well as repeatabilities. The second objective for this study is to establish reference samples and data sets so that we can use it for the validations and benchmarking for the communities. And finally, we like to also build a unified framework for PCR sequencing analysis to train FDA review internally and also use for the evaluation of the supporting data by sponsor. So we start a pilot study, which is very simple design that's using two cell lines, Dowdy and CA46. We mix them with the design ratio like 3:1, and then we put the mixed dilute into PBMC at 10% and 1% dilutions. We then run a series of the PCR-seq assays and also single-cell sequencing. And from some of the assay, we perform some QC and looking for basic the performance in terms of the alignment of the reads on the PCR-seq. And you can see that three assay providers, they have different QC characteristics. That's just one of those QC metrics we can use to looking for the performance. We also are looking to a series of PCR lines so that we can choose them to determine as a candidate to develop the reference material. So this is the 50 cell line PCR lines. We went through the three different assays with the PCR assay from New England Biolab and also from the Tegra Bio. We also run the whole transcriptome sequencing on each cell lines. And with all three assays, we determine the clonal type for each cell lines. And then eventually we'll be able to identify a set of cell lines that are pretty much monoclonal, i.e. they only exhibit a single clonal type for heavy chain and light chains. And then this evidence actually coming unambiguously coming from three assays. So those cell lines is a good candidate for those cell lines are good candidate for us to develop reference material for the benchmarking study. So besides we do the pilot study, we also conduct the in silico study to serve as a proof of concept. Basically, we generate the artificial reads. Actually, we take reads from the pilot study as well as from 50 cell line study, and we mix them by purpose. So we take the 9 cell line that we know is unambiguously is a unique clonal type, and then they have different representing different V segment genes by each cell lines. So we take reads from each cell lines and then mix them at a different percentage, right? So we have series mix of 9 cell line at different percentage. And then we take this master mix of reads, dilute into reads from PBMC at 10%, 1% tier to 0.01%. So we have series dilution, 4 series dilutions. And we repeat this multiple times so that we make sure it's random, the mix and dilute. And from this synthetic reads and dilutions, we can observe, the detection sensitivities in terms of the the clone type fractions, and then with the expected value versus observed value, right? So this is original master mix for the heavy chain, and the lower panel is light chain. You can see for the master mix of the heavy chains pretty much in line with what we expected, and so did the light chains. But when you look at series dilutions, here to like the lowest dilution to 0.01%, you start to see the deviations away from the observed values, which is what we expected, because with lower fractions of the clonal type, your detection's capability is dropped. And then we also look at the detection sensitivities in terms of the risk coverage or UMI coverage. And based on that, our preliminary result from in silico studies showing that with about 100K UMI coverage, the detection limit is around 10⁻⁴. And this actually providing guidance to us to play in a real experiment. We would like to increase the risk coverage to up to 1 million UMI, so that we can have fully the examination in terms of risk coverage and also detection sensitivity. So let's come back to the basic study design for the full study for this project. So basically, we will have starting with 50 cell lines coming from DSMZ, and then we defined the cell lines and pick up 9 to 10 cell lines and making master mix, and then generate reference material, and using this reference material to perform benchmarking study against different assay. Let's say we have collected 10 PCR assay currently dominate on the market. We also can benchmarking different sequencing technologies. So far, we have been identified 6 major sequencing platforms on the market. And also we want to looking for the latest technology for the single cell sequencing as well. Besides those, the wet lab and tools, we also like to evaluate bioinformatics tools in terms of their impact for the data analysis and also filtering of the reads. Finally, we would like to conduct clinical validation. That is, we like to using at least 2 PCR sequencing technology and work on the same sample set of the hairy cell leukemia. And we'll be using this comparison, looking for the performance of different PCR sequencing technology in terms of MRD biomarker discoveries. So the logistic flow for this study is also quite straightforward. We basically received the cell line from DSMZ. We start to cell expansion, generate the DNA, RNA, and make the master mix. And then we distribute the DNA, RNA as well as cell to the assay provider or sequencing provider. And then we collecting data on the preceding FDA where we do centralized analysis. So far, we have been finished the generating the materials, and then we also finished the QC on the materials, and the result look very, very good and then satisfactory. And so we will start to distribute materials among our working group member, and then we expect to finish the all experiments by the end of this year. So here's a list of the participators for this study. So basically, it's a kind of consortium type of study, including our government agency from FDA centers, as well as the NCI, VRC. We also have a set of the research university and those from the communities focus on the PCR sequencing. And also we have participated from the sequencing platform provider as well as the assay providers. So this is a collection of the expertise from regulatory science, immunology, as well as sequencing technology, and also bioinformatics. So far we have over 100 scientists from 40 plus institutes. So with that, I would like to conclude my presentations and thank you for your time.
Thank you, Dr. Wenming Shah. That's an interesting talk. And thanks for sharing your data on sequencing technologies in B-cell receptor-based immunome profiling. I guess in the interest of time, we had to move on to the next speaker. The next speaker is a colleague of mine from NCTR, Dr. Sangeeta Khare. She's been working on microbiome for a while, and her talk today is titled Host Microbiome Crosstalk, Disruption of Gastrointestinal Barrier as Toxicity Assessment Tool. Sangeeta, take over.
Thank you. Can you see my slide and hear me well?
We're seeing the presenter view of your slide currently. Would you like me to share instead?
No, I will.
Yeah, if you choose swap, there we go. Perfect.
Is that all right?
That's correct. Thank you.
Okay. And you can hear me well?
Yes, we can.
Okay. Thank you, Manju, for that introduction and the invitation. So before starting my presentation, I will just like to read this disclaimer that finding an opinion presented here are my view and does not reflect FDA Food and Drug Administration. And also mention of any commercial product is for the clarification only, and it is not endorsement for the use of it. So with that, I'd like to focus today on the gastrointestinal homeostasis and how it plays role into the health and disease. This homeostasis can be changed by several xenobiotics, and that includes some of the FDA-regulated products. Usually, toxicology issues that needs addressing for the FDA-regulated products include no adverse effect observed, an acceptable human daily exposure, then possible developmental and reproductive toxicity, factors that affect different responses by subpopulation, and also the potential targets organ of toxicity. So, however, among the organ toxicity, the endpoint for the gastrointestinal toxicity are still not well defined. But FDA has recognized microbiome research as one of the focus area for the regulatory science. And today, that's what I will be basically covering. So if you can see that gastrointestinal tract homeostasis is maintained by the microbial barrier as well as the host barrier. And my talk will cover like combination of both into the toxicological assessment, because if we see the host barrier, the epithelial cells and the gut-associated mucosal responses play a major role into the homeostasis. And disturbance in homeostasis can lead several outcomes that include dysbiosis, immune activation, malabsorption of nutrition, and extraintestinal effects, etc. So in our lab, we have been using xenobiotic microbe interaction tool as well as the xenobiotic host interaction tool using animal and non-animal model. And we also looked the effect of the host and microbe onto the xenobiotic metabolism too. And towards this, we have been using several tools, and they are based on the omics approaches that include 16S sequencing, metatranscriptomics, then gene expression analysis, proteomics, metaproteomics, and also the metabolomics. And in our lab, we have been studying several xenobiotics that include environmental pollutant, additives, nanomaterial, and antimicrobial residues. Apart from this, we have been also studying the gut-liver axis and gut-brain axis using a special animal model. Those are collaborative cross animals or the transgenic model. So today I will be giving the snapshot for the triclosan results, then one of the nanomaterial, that is nanocellulose, and one of the antimicrobial, that is aloin. So I'll start with the triclosan. Its impact first, I'll give introduction. So it has been present, it had been present in several of the products that were used on a daily basis, but several federal agencies have banned the product, but still in some countries, like in Canada, 0.3% concentration is still allowed into the products. So question is whether there are any developmental effect of the triclosan. To address this, we have used the rodent model to mimic human developmental exposure. And for this, we collaborated with the National Toxicological Program and NIEHS and conducted this study. In this, the adult animal at GD6, they were gavaged with triclosan with 3 different doses, 100, 500, and 1,000 milligram per kg body weight. And then during the birth, they were still kept with the moms, and they got exposure through lactation. And at PND 12, they start gavage. The animals were, or pups were gavaged separately too. And we collected the samples from dam at GD18, PP7, and baby 28, and from pups at PND 28. And we looked for the 16S sequencing, triclosan detection, immunoglobulin A, and also the metatranscriptomics, so that if bacteria is there, what they are doing actually. So our first result shows the analysis at the phyla level. And here we can show that at GD6 itself, we can see at high dose, there was high abundance of the proteobacteria that you can see here. And during the exposure at PND 28 also, we can see that proteobacteria or the pathogenic bacteria was present into the gut of these animals. Then we went ahead and did the genus-based analysis. And here you can see a clustering that controlled gavage. All those animals were together, like came out together. And then when we look for the TCS 100 or 500, we can see a time and dose dependent gradual shift into the abundance of the bacterial population. So we can clearly say that during the gestation and early developmental stages, we can see changes into the microbial diversity and species richness. Then because immunoglobulin A is a potential regulator for the commensal microbiota, so we looked at levels of that, and we can clearly see at postpartum 7 and 28 days, we can see that there was significantly high levels of IgA, and it correlated with the bacterial abundance also. And then we looked, okay, these bacteria are present, what they are doing. So we did the metatranscriptome analysis. And what we were able to see here, that TCS effect can be seen at GD18. There were several human disease-related genes. Those were expressed at GD18 level itself. And at higher dose, as you can see here in the blue and pink representing male and female pups, we can see higher abundance of the gene related to the disease, and it was more prominent into the female as compared to the male. And then we were also able to see dose and sex-dependent metabolism-related pathway, as you can see in the third graph, that we were able to see more metabolism-related gene, and that correlated with the accumulation of the TCS into the female pups. So with this, we can say that metatranscriptome analysis and the 16S analysis clearly showed a significant changes in the functional gene during gestation and early development upon the triclosan exposure. So our next study was in collaboration with Harvard School of Public Health in a study while using the nanocellulose, they found that it reduced the intestinal digestion and absorption of fat. So then the question was whether it is related anything with the gastrointestinal tract. So our study then was conducted and in which gut microbiota composition and functionality changes were assessed. So as per the classical biochemistry, we know triglyceride gets hydrolyzed into the intestinal lumen and it gets resynthesized into the intestinal mucosal cells. So we studied both host as well as microbiome responses. So for this, rats were gavaged with the nanocellulose in the presence or absence of the cream and the water gavage. And then we looked for the microbiota, intestinal permeability related gene, and the mucosal cytokine immune responses. So here we can see a clear distinction for the microbial diversity. These two are the control gavage, and here these three panels are the gavage with the nanocellulose, cream, or cream and nanocellulose. Similarly, when we looked at the species level, we can see a clear differences. The clustering was clearly different, and the heat map analysis showed that there was clear segregation between the experimental group. So then we looked what were the significant species that were present. And here we can show you some of the species that were significantly different as compared to the control. These significantly abundant species were involved into the production of large quantity of the short-chain fatty acid, promote IgA secretion, elevated insulin level, and degraded the cellulose, and also they were also involved in the consumption of more carbohydrate, especially fiber. And there was lower abundance of the bacteria. Those are involved for the pathogenic bacterial species. So next we look whether there are any changes into the intestinal permeability. So we were really not able to see any significant changes or higher expression of the gene. Rather, there was lower expression of the permeability related gene. That means there is no disturbance into the intestinal mucosal permeability. Then we looked about the cytokine profile into the ileal mucosa, and there was activation of the cytokine cluster that is involved into the promotion of the proliferation of naive CD8 cells. So you can see IL-6, IL-10, 7, 18, and EPO were significantly up into the cellulose nanofiber. So with this, we came with a mechanism that is described here, that at the gastrointestinal tract, due to the nanocellulose, there is presence of the or abundance of the microbes. Those are involved into the beneficial effect. Cellular integrity was not disturbed that much, and also the cytokine profile was helping the host. So probably that could be the mechanism at the gastrointestinal level for the action of the nanocellulose. My next study is about the aloe vera study. And in this, we have used actually the in vitro or non-animal model. So aloe vera, we know that it is used to as a very popular consumer product. But an earlier study at NCTR had showed that there was increased incidences of the adenocarcinoma when they were given to the Fisher rats. So we wanted to see what could be the molecular mechanism behind this abnormality. So for this, we have used in vitro bacterial cultures. We took specific common cell bacteria, incubated that with allowing different concentrations of allowing, find the MIC or minimum inhibitory concentration for these. Then we also looked to the, because these were only the selected bacteria, so we looked the in vitro fecal slurry and incubated that with allowing and performed the 16S sequencing. And for the host side, we looked the intestinal permeability responses using the colonic epithelial cell lines. And what we found that at the microbe level, there was a decrease in the live bacterial culture population when we incubated it with the fecal slurry was incubated with the alloin. And then we also found that the low abundance of deuterate-producing bacteria were present, and that also was correlated with the short-chain fatty acid production that was reduced in these in vitro exposure. And for the host side, there was a disruption into the intestinal barrier integrity and increased cell proliferation and inhibition of the pro-inflammatory cytokine. So probably this could be the mechanism interaction of these that may lead to the hyperplasia and the adenocarcinoma formation. So we have also collaborative project in which we are looking the effect of extraintestinal effect of different diets. For this, we are using the collaborative cross animal to see high-fat, high-sucrose diet, and the transgenic animal that are prone to the Alzheimer's disease. We are looking the time-dependent effect or longitudinal effect with the age on the gut microbiota and intestinal permeability, and also looking effect of the high-fat diet on these animals. And with all these studies, finally, the question is what we have learned. So now we have made a decision tree for the evaluation of the gastrointestinal toxicity. So if any xenobiotic is there, we would like to see, okay, if the minimum inhibitory concentration of that xenobiotic is already known. If it is known, we go to the animal study. If it is not known, we start from there and then go into the animal study if there is any microbicidal effect of the xenobiotic. And then from these animal study, we go for various, like whether residual exposure, environmental or occupational exposure, acute exposure, or the developmental exposure. And if we find the changes here, then we go for the who is there and what they are doing to answer those questions using the omics approaches, and then try to correlate to different omics to see how metabolism of the xenobiotic is impacted and how xenobiotic is impacting the gastrointestinal tract. So with this, we have tried to answer the toxicological issue that need to be addressed FDA-related product at the gastrointestinal tract to see various concentration, like minimum inhibitory concentration, to show the effect of the xenobiotic. And we have defined the ADI or the dose level at the systemic exposure, then potential toxicity at the organ. Then we have also shown developmental and reproductive toxicity using the transgenerational study, and factors that affect different responses by subpopulation. We are currently doing collaborative cross animals and transgenic animals. And how this helps for the translation and regulatory science. These results will lead to the modernized toxicology to enhance product safety. And we are developing tools to determine how gut microbiota composition and functionality changes. And most importantly, dissemination of these findings will empower consumer to make better and more informed decisions about their diets and the health. And with this, I would like to thank my team. There are several people involved in this. Today, I have presented work. Those are in the red font. And the green font, actually, we have two posters, one on the transgenic animal and one in collaboration with Dr. Kupan Gokhlan on the nano drug. So they are also in FDA website. So if you're interested, you can go there. And thank you for listening.
Thank you, Sangeeta, for an excellent talk and very informative information on host microbiome cross interaction. With the interest of time, we need to move on to the next speaker. And attendees, please, if you have any questions, type into Q&A box so we can discuss this at the end. Our next speaker is Carolyn Young from CBER, and she's going to talk about the regulatory perspectives on advancing regenerative medicine products and emerging technologies. Carolyn.
Thank you. Can you hear me and see my slides?
Yes.
Great. Okay, thank you so much. Good afternoon, everyone. We appreciate you tuning into the last session of the FDA Science Forum. I'm Carolyn Young in the Office of Therapeutic Products in the Center for Biologics Evaluation and Research here at FDA. And I'm going to take this plane a few thousand feet up, and I'd like to thank the organizers for inviting me to provide, an overview of some of the agency's perspectives on regenerative medicine products and related emerging technologies. So as you've probably encountered, regenerative medicine has a lot of definitions in the field, and surprisingly, regenerative medicine therapies weren't defined in law until 2016. From a regulatory perspective, regenerative medicine therapies include cellular therapies, tissue engineered products, certain human cell, tissue, and gene therapies, as well as xenotransplantation and combination products using such therapies. Here are several examples of products that would meet the definition of a regenerative medicine therapy. So FDA regulates regenerative medicine therapies, and I'll run through the who and some of the how. There are 3 human medical product centers at FDA identified in the red square. And in CBER, there are 3 offices responsible for regulatory review of products under CBER's purview, and we'll be focusing on the Office of Therapeutic Products, or OTP. OTP regulates a really diverse array of products. Some classes of products with more specific examples are provided on this slide, and notably, many of the products on this slide would be considered a regenerative medicine therapy. So to review such a diverse array of products, the Super Office of Therapeutic Products, formerly known as the Office of Tissues and Advanced Therapies, was organized just this past February, as shown in this slide. And it should be noted that within each of the product regulatory review sub-offices, the 3 on the left side, there are research-focused labs investigating regulatory science issues relevant to the products that we regulate. And I'll touch on those later in the presentation. So now I'll transition to regenerative medicine product regulatory review. Regenerative medicine products can restore, repair, replace, or regenerate, and these are incredibly complex processes. So accordingly, regenerative medicine products are complex in nature. For example, you can have a combination product consisting of a device scaffold and biologic constituent consisting of cells to build a tissue engineered construct. So on the right side of the slide, are 2 approved tissue engineered products, the top for repair of cartilage defects, and the bottom, a cultured skin graft. So such products are very innovative and often undergo tweaking during clinical development. The complex nature of these products is also reflected by the regulations that can apply to these products depending on what they're comprised of. For instance, biological products are also drugs, so the biologics and drugs regulations apply. But if there are human cells and or devices incorporated into the product, the respective set of regulations will apply. So regulatory review, whether it's in the pre-market investigational stage or conducted post-marketing authorization, is really driven by benefit risk assessments, informed by evolving science, technology, medicine, policy, and regulatory requirements. So throughout this forum's sessions, there is a common thread, which is data-driven decision-making. So you have a flavor of the diversity of regenerative medicine products, but what is especially challenging for regulators is that regulatory review and benefit risk assessments are highly product dependent. The scientific novelty of regenerative medicine therapies can present unique challenges for meeting regulatory requirements. So introduce new manufacturing modalities and emerging technologies. In these cases, being data-driven is challenging when there's not a lot of data or experience to draw from. So here are just a few examples of product-specific considerations. For example, scale. So a single lot of products, like small molecule drugs, could treat 1,000 patients or more. Many regenerative medicine therapies will have a lot of one, meaning one product manufactured for one patient. So this can also introduce testing challenges because producing one of something may limit being able to test and needing to test through non-destructive methods, many of which are not yet available. So we encounter lots of issues with variability as well, since a lot of regenerative medicine products source materials are patient specific. And, when one uses one's own cells to produce a product, there is inherent variability. So I mentioned previously that regenerative medicine product development can be iterative. So it is imperative that sponsors and the agency interact frequently during the product life cycle. So you can see here that there are opportunities at all phases to discuss product-specific development with the agency. And just for your awareness, there are also a number of programs available, including one tailored just for regenerative medicine therapies for serious conditions that can expedite both development and availability to patients if the promising treatments meet the eligibility criteria for one or more programs. Regenerative medicine therapy products are generally cutting edge, and a variety of advanced technologies are being implemented in their development. And one area of focus for the agency is advanced manufacturing. An earlier session in the forum was dedicated to this pretty broad topic. So I'll just provide, highlight some key points. So here are just a few benefits, for example, of integrating novel technology and their approaches to product manufacture. So there is a lot of incentive to develop novel technologies that can, for instance, automate heavily manual processes like cell culture and do so in a closed system. So what if you're a developer of an advanced technology intended to advance the development of CBER-regulated products? CBER has a dedicated program established to interact with developers of advanced technologies. Here are some highlights of the CBER Advanced Technologies Program, which has a dedicated team, or CAT, and the program are critical to building internal scientific and regulatory expertise. Because our decisions are data-driven and informed by experience, an early introduction to cutting-edge technologies provides CBER with invaluable information and time to discuss regulatory challenges and implications emerging technologies may present. And here are some specifics on the CAT team I mentioned earlier. The CAT team consists of representation across most of the offices, regulatory review offices included, and leadership within the center, drawing in relevant subject matter experts, policy, and compliance staff as appropriate. So if you're wondering what some of the examples of advanced technologies we've encountered, this slide just provides a flavor of what the CAT team has seen on the not too distant horizon. So we have continuous manufacturing, fully closed, automated, scalable, and remote controlled systems for cell therapy products, cell lines, genome editing, artificial intelligence is huge, as well as multi-product manufacturing facility design. Less well known is that CBER promotes development and adoption of advanced technologies for the manufacture of CBER-regulated products through extramural funding. Such research is critical in addressing knowledge and experience gaps that we identify with emerging technologies. Briefly here, the agency has been working to establish a regulatory framework to address current and potential future manufacturing innovation for CEDAR and CBER-regulated products through its FRAME initiative. discussion papers regarding distributed and point-of-care manufacturing and artificial intelligence have been published, and accompanying workshops convened or upcoming. An FDA co-sponsored workshop on the regulatory framework for the utilization of artificial intelligence in pharmaceutical manufacturing will be held this fall. So I'd be remiss if I didn't touch on regulatory science, which is at the heart of clinical translation, our regulatory decision-making, and standards development. And many tuned in here today are engaged in regulatory science research here at FDA. And with respect to regenerative medicine products, we have research looking into the impacts of the iterative product development process and less well understood aspects of our products, such as their mechanism of action. So here I've listed our cellular therapy research and respective PIs. Our research groups are tackling some of the key challenges we encounter with cellular products, with goals that will inform our regulatory decision-making for such products. For example, gaining a better understanding of how cells interact with their microenvironment and what happens to cells once they're administered to a patient. And hopefully many of you were able to catch my colleague, Dr. Kyung Sung's presentation yesterday, which talked about advanced analytical methods for assessing efficacy of regenerative medicine cellular products. We also have labs focused on gene transfer and immunogenicity research. And now I'd like to say a few words about standards development, which can really streamline development and regulatory review of FDA-regulated products. OTP, including our research labs, are also engaged in standards development. So CBER encourages the use of appropriate standards in the development of CBER-regulated products, and the use of voluntary consensus standards can assist stakeholders in more efficiently meeting regulatory requirements and increase regulatory predictability for regenerative medicine therapy products. CBER is upstanding a recognition program for standards relevant to the development and review of regenerative medicine therapies, and with hopes to promote the development of standards in this field. Standards are, very prevalent, especially in the field of medical devices, but, we really could use more standards in the regenerative medicine therapy space. And I'll end my presentation by putting in a plug for the Regenerative Medicine Fellowship under the CBER Service Fellowship Program. I don't have time to go into the details here, but encourage you to check out the webpage for details. Essentially, the fellowship aims to enhance FDA's ability to regulate regenerative medicine products and support the development of highly trained scientists and engineers with unique multi-center experience. So here's my contact information and some additional resources, and thank you again for tuning in.
Great. Thanks, Carolyn, for an interesting talk and sharing CBER perspectives on regulatory issues and advancing regenerative medicine products and emerging technologies. Thank you. Our next speaker is Dr. Nahid Kamal from CEDAR, and she is presenting an interesting talk entitled Dermal Drug Delivery via Dissolvable Microneedles, Formulation Variables Affecting Critical Quality Attributes. Dr. Kamal.
Yeah, thanks so much, man, for the nice introduction. I hope you hear me, right, and see my presentation?
Yes, we do.
Okay, wonderful. Good afternoon, everyone. I'm Nahid Kamal. I'm a pharmacologist, reviewer, and a principal investigator in Office of Testing and Research under OPQ of CEDAR FDA. Today, I'm going to present one of our ongoing research work, which mainly focus on our understanding on effect of formulation variables on the critical quality attributes of the dissolving microneedles. So in next few slides, I will talk about microneedles, manufacturing of microneedles and challenges, as well as various characterization results to understand the critical quality attributes. Before going into details, here is my disclaimer. This presentation reflects the views of the authors and should not be constructed to represent FDA's views or policies. So ensuring pharmaceutical product quality is very important to us because As per FDA's definition of pharmaceutical quality, a quality product of any kind, it could be computer, car, or cell, etc., consistently meets the expectation of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuming every dose is safe and effective, free of contamination and defects. It is what gives patient confidence in their next dose of medicine. Now I will give you an overview on the microneedles so that you can get familiar with this technology if you haven't yet. Microneedles are micron-sized needles used to deliver drugs into the skin after bypassing the stratum corneum, which is the outermost layer of the skin and limits the drug absorption through the skin. Due to the innovative nature of this delivery system, no pharmacopoeia standards currently exist and no approved drug product in the US market yet. Therefore, there are various types of microneedles. Among them, dissolving microneedles are manufactured from biodegradable polymers to deliver drugs in a simple, invasive way. After insertion into the skin, the microneedles dissolve in the surrounding interstitial fluid of the skin and release the drug. So development of any kind of microneedles with desired critical quality attributes, especially mechanical properties that ensures skin insertion for adequate dosing, remain a challenge. Therefore, the main objective of this work was to understand the effect of formulation variables, namely, in our case, is polymer to API ratio that we have investigated on the various critical quality attributes of the dissolving microneedles. For example, microneedle formation, topographical properties, mechanical characteristics in terms of needle breaking strength, skin insertion ability, and as well as the drug release. For understanding the effect of formulation variables, we have manufactured 3 different dissolving microneedles, as you can see here, named as F1, F2, F3, by varying polymer to API ratio. For this purpose, 3 different casting solutions were prepared by varying concentrations of 10 to 14% of polyvinyl alcohol, or in short, PVA as polymer, while keeping the API concentration fixed at 15% concentrations. In this study, we have used metoporous as a model drug. For manufacturing the dissolving microneedle, we have used the PDMS mold that was designed to deliver 10 by 10 or 6 by 6 dissolving microneedles with pyramidal shape and height of 600 micrometer, and base diameter of needle is 200 micrometer. So I'm not going into details because of the time restrictions of the manufacturing process. In short, the overall manufacturing process involved application of the casting solutions onto the mold, and then this mold goes for centrifugation. And after centrifugation, we add a blank formulation on top of that, on the base of the needle to form a backing membrane. And after that, we put the system into the oven for drying, so that we can get the dissolving microneedles area at the end of the drying. At the end of the drying, after separation of the dissolving microneedles from the mold, these microneedles subjected to the characterization studies. The first characterization that we do the microscopic analysis to see the formation of the needle. I'll be talking about it in the next slide. One important thing, since we are talking about the polymer to API ratio, so the final PVA to drug ratio in the dissolving microns that we have manufactured, 3 different microneedles were 0.67, 1.33, and 2.67 for F1, F2, and F3 microneedles respectively. During manufacturing, we came up across various challenges. A few selective challenges are provided here. For example, we have observed that the viscosity of the casting solution, especially 40% solution, impacted the filling of the microcavity of the mold, resulted in incomplete formation, which is shown in the upper image in the slide, which further resulted in poor yield. Then another very common observation that we had a low drop recovery from the dissolving microneedles due to significant amount of the drop loss during centrifugation. I believe most of people who are familiar with these drawbacks of these dissolving microneedles, whoever work in this field. So this drop loss further caused us the analytical challenges during the drop quantification. So in next few slides, I'll be talking about the results, our findings, and then how this impacted the critical quality attributes. As I mentioned, the first characterization that we have performed was macroscopic examination of the macroneedle formation, because if the macroneedles did not form, then there is no point to subject them for the further characterization. So the result of optical macroscopic examination show all three dissolving macroneedles, array of F1, F2, and F3, in most of the cases form completely with sharp tips, as you can see in the image of A for F1, B, and C for macroneedle F2 and F3 respectively. Image D showing the close view of the needles at higher magnification. The percent recovery per manufactured batch was kind of low, like 75% to 88%, regardless of the various compositions. It was due to a few failure cases that we have observed during the macroscopic analysis. For example, as you can see in the lower panel of this slide, the images, presence of bubbles inside the needle or in the backing membrane, as shown in the image D, and broken needles or separated base as shown in image E, tilted needle showing in image F. And one of the very commonly defects that we have observed, slightly bent tip as shown in the image G. This defect is very common, we have observed. So, however, only completely formed dissolving macroneedle array free from any kind of defects, so we're subjected to the further characterization testing. Then we have looked at the topographical properties of the needle by using the artificial intelligence assisted image analysis. This work was performed in collaboration with DGM. The results show that the macroneedle formed completely, having pyramidal shape needle with consistent height, with consistent tip circular diameter and cross-sectional base diameters. The outer surface of the needles are smoothly formed without any defects. Then we also have investigated the spatial distribution or API localization in the macroneedles using same AI-assisted image analysis. The result of intensity versus the distance curve, the distance meaning from the backing membrane toward the needle tip, and the grayscale image processing as shown in this slide show that the AI grayscale intensity varied strongly from backing membrane toward the needle tip in F2 macroneedles, and modest variation was observed in F3 macroneedles, while F2 placebo shows little to no variations. These results suggest that localization of the drug more in the needles of F2 dissolving macroneedle than in the F3 macroneedles related to the placebo. The results also reveal that drug was uniformly distributed in the macroneedles array in a dissolved state. No separation was phase separation was observed. We couldn't add the result of F1 macroneedles because the analysis is still ongoing. One of the important characteristics of the macroneedles is the mechanical properties of the needle. So we have investigated this property by using texture analyzer at compression mode. So the results show the results showing the right side of this curve. So the results show that the breaking strength of the needle, the breaking force per needle was increasing proportionally and significantly from 0.13 Newton to 0.35 Newton per needle, as the PVA to API ratio was increased from F1 to F3 respectively. The breaking strength of the placebo needles were also increased, as you can see in the figure, as a PVA to drug ratio are increased and found stronger than the drug-loaded needles. These differences may be ascribed to the difference in API concentrations, that is the decrease in the API concentration in the drug-loaded needle or absence of API in the placebo needles that may act as a plasticizer in the macroneedles. Another very important mechanical characteristics of the macroneedle is skin insertion ability of the needle. We have used human cadaver dermatomy skin for this test, since skin is considered golden standard for any kind of skin-mediated drug delivery system. So needle was applied on the skin either by thumb force or by texture analyzer at compression mode. In both cases, results show all three dissolving macroneedles were able to pierce the skin completely. Images are shown in the top right of the slide. The first record to pierce the skin around skin thickness of 500 plus minus 600 micrometer thickness was found that the force was decreasing significantly from 0.08 to 0.03 Newton with increase in PPA concentration, which was, the good thing is, which was lower than the breaking force of the needles as I described in the previous slide. But I must admit, when skin thickness increased above 600 micrometer, no correlation was observed in terms of change in formulation composition. It may be ascribed to adequate hardness, suitable shape and sharpness of our needle tips that were able to overcome the various viscoelasticity of the skin. Now, I will be talking about various release testing methods that we have investigated, because based on the geometry and as you know, dissolving microneedles has a drawback of low drug dosing. So because of that, it was very difficult to find a suitable release or the dissolution method that can be used to evaluate the drug release from the needles. So this is why we investigated various methods. The first method was we have investigated that immersion technique, where it's shown in the left side in the slide. In this method, a dissolving macroneedle placed onto a vertical diffusion cell containing PBS as release media, and the sample was withdrawn at various time points up to 1 hour, then analyzed by validated HPLC method. Results showing in the right side figure in this slide that the overall drug release was decreasing proportionally and significantly by decreasing the PVA to API ratio. The highest drug was achieved from F1, followed by F2 and F3. It is important to be noted that the needle dissolved very fast within 15 to 25 minutes related to the backing membrane. The overall method was found discriminatory. The next method that we have investigated was agarose gel film, where a dissolving macroneedle was applied on 10% agarose gel film. As you can see the image in the left side of this slide. The macroneedle was held for various time points onto the gel, and then removed it to quantify the drop from the gel at the end of the various holding time to observe the needle deformation as well as to quantify the drug release from the gel. Results show the needles were dissolving over time for all three formulations, and as you can see in the images, at 1 second, 15 seconds, 30 seconds, or 60 seconds, or 1 minute. However, the drug release data show non-discriminatory effect on the formulation changes in the needles or in terms of the drug release or in terms of the needle deformation. Then similar way, we have also investigated the drug release by using human cadaver dermatomy skin at non-sink condition. But for this experiment, we have held the dissolving microneedle area on the skin from 1 minute to 30 minutes, and the drug release was quantified from the skin after each holding time. And same to needle deformation also observed under microscope. The results show the needle were able to dissolve at various time points, as you can see in the images on the right side of this slide. However, like agarose gel method, this method was also unable to detect any changes in formulation variables in the needle. So this could be attributed to the limited volume of the moisture present inside the skin at non-sink conditions. So therefore, this method also found non-discriminatory. Lastly, we have also investigated drug release using synthetic membrane, where macroneedle was inserted onto a synthetic membrane that was fitted to a diffusion cell. The experimental condition kept same as the immersion technique. The results show simultaneous release of drug from the needles and the backing membrane due to back diffusion of the release medium into the membrane through the holes that was created after insertion of the needles into the membrane. So resulted in uncontrolled drug release. So I believe this is the end of my discussion. So the key understanding or the key summary of this presentation is like polymer to drug ratio affects mechanical strength and drug release, but it does not affect skin insertion. AI-assisted image analysis unveils spatial distribution of the drug with acceptable resolution. Among various release testing methods investigated, agar's gel, synthetic membrane, or skin at various holding time showed no discriminatory power for the formulation changes in the needles. These methods need further development. The immersion method was found discriminatory and showed the effect of PVA to APA ratio on the drug release. Hence, we believe this method can be used during product development. Our future study will focus on the drug release from the dissolving microneedles as a function of time, employing human cadaver dermatomy scheme and employing vertical diffusions as unobserved conditions. With that, I would like to acknowledge following people. I believe without their support and collaboration, the outcome of this research would not be as successful. Thanks to Samuel Akampong, Rukun Zaman, Ahmed Zadan, Mohammed Ashraf, Thomas O'Connor, Karen Stenzer, James Norman, Tammy Ewing Tang, I'm sorry. Then I'm also grateful to Oak Ridge Institute for ORI's support, Chief Scientist Course Intermore Grant for partial financial support, and Andrew Clark and his team for the collaboration with us for AI-assisted image analysis from DGM. Lastly, I would like to thank to FDA Science Forum for giving me this opportunity to present our work, and of course the audience for their attention. So if you have any question or any feedback, please hold it until panel discussions. Thank you.
All right, thanks, Dr. Kamal. It was a very interesting talk on dissolvable microneedles and dermal drug delivery. In interest of time, I got to move on to the last speaker. Of course, there are questions we will discuss or go over it at the end. So the next speaker, the last speaker, of course, not the least, comes from CDRH. And my apologies if I butcher or mispronounce the name. It's Xian Chow.
Yeah, that's right.
Awesome. Thank you. Appreciate that. And the title of his talk is an assessment of trabecular bone stiffness using radiomics and deep learning features. All right, take it over.
Thank you. Good afternoon. I'm from the Office of Science and Engineering Laboratories at CRH, where I lead a project on quantitative imaging biomarkers of bone health. So today I'm going to introduce some ongoing research here. So first, some background. This work is about the assessment of osteoporosis. It's a disease characterized by the loss of bone mass and increased fracture risk. It's prevalent and costly, impacting 10.2 million US adults, 80% of whom are women, and is responsible for 2 million fractures and $19 billion in related costs annually. So currently, osteoporosis is assessed in two ways. The flowchart on the right shows a management algorithm for assessing patients at risk of fracture. FRAX is a risk calculator based on clinical factors and estimates a 10-year fracture risk. However, the diagnosis of osteoporosis is typically made with imaging to determine bone mineral density, or BMD. This is either through dual energy X-ray absorptiometry or quantitative CT. So during an initial assessment, if the fracture risk is deemed intermediate from clinical factors, then additional imaging might be prescribed to determine BMD. So bone strength and fracture risk is not only dependent on the bulk density of bone, but also how the bone structures are arranged, that is the morphology and microstructure. And if we want to make measurements of microstructure, we need to take a look at image texture. One of the FDA-cleared algorithms for texture analysis of bone is the trabecular bone score, TBS. This is a texture-based biomarker based on the variogram of a 2D DEXA projection image. We can also compute texture features from CT images, while the spatial resolution of a typical QCT scan limits the extraction of texture features. Recently, there are new and emerging imaging systems increasingly capable of visualizing bone microstructure. This includes specialized orthopedic CT systems, such as the HR-PQCT, high-resolution CT, photon counting CT, and so on. On the right, I have included examples of distal extremities imaged with HR-PQCT and temporal bone imaged in photon counting CT. And there has been multiple clinical trials to evaluate the relationships between microstructure and fracture risk based on CT. One such example is the Offaly study, where more than 1,000 postmenopausal women were evaluated for 10-year probability of major fracture. The fractures included fragility fractures of the wrist humerus, vertebrae, and hip. The volunteers were evaluated with FRAX, DEXA, and HR-PQCT. Mechanical analysis were performed on the HR-PQCT images to compute the stress and strain maps, such as the analysis shown here in the middle. And for this cohort, they found that some bone microstructure parameters in HR-PQCT images, such as trabecular bone, BMD, trabecular numbers, and connectivity, were of good prediction power to predict fragility fractures or incident fractures. So the goal of our project is to develop texture-based features that can be measured from high-resolution CT and used to estimate bone quality. In turn, this may lead to improved screening and more streamlined drug trials. For this study, we're looking at the stiffness of trabecular bone as a surrogate to fracture risk. We hypothesize that compared to using BMD alone, which is the current standard of care, the use of texture imaging features can improve the estimation of trabecular bone stiffness. Clinically, this assessment might be carried out for patients with high-resolution CT scans, where the regions of interest may be selected on the lumbar spine. Texture features can then be extracted and used to generate a stiffness or fracture risk prediction. So next, I'm going to briefly introduce some of the texture features we're looking at. In this work, we consider 3 sets of features shown in this figure as A, B, and C. They are radiomics, gradient structure tensors, and deep learning features derived from ResNet. We also consider different combinations of these features when used in estimating bone stiffness. Note that bone mineral density, BMD, which is the current standard of care, is included as one of the radiomics features that corresponds to the mean value in a bone region of interest. All features are input into a random forest regressor to predict and train to predict bone stiffness. So next, we're going to go into a little bit more detail about what these features are. Radiomic features are engineered texture features used in diagnostic radiology or radiotherapy for prediction and prognosis. An advantage to using radiomics features is that these features are mathematically defined and very explainable. So these features might include first-order features such as mean, standard deviation, percentiles, as well as more advanced patterns such as the grade level correlation matrix, the GLCM, which describes the frequency that a voxel with a certain value neighbors a voxel with another value. So for our analysis, a total of 92 radiomics features were computed using the open source software PyRadiomics. The plot on the right shows the relative importance for different features when used in a random forest regressor to estimate bone stiffness. And from this distribution, we can see that in this specific case, a subset of 20 features out of 92 contributed most to stiffness prediction. Now onto our second feature set. Trabecular bone is anisotropic with large plate-like structures oriented in the load-bearing direction, as shown in the image of the femur head. So we consider using features which are direction dependent. The gradient structure tensor, or GST, can be applied to determine the orientation of trabecular bone. An example of such analysis is shown by the red arrows overlaid on the femoral head. Here we use GST to determine the principal directions for our bone ROIs. The 3 principal directions were derived using the 3 eigenvectors of the GST structure tensor, and that's shown on the right here. So the angles between the principal directions and the loading directions were used as 3 direction features. So our final feature set are features derived from a neural network. Here we used a residual neural network known as a ResNet. This architecture is quite popular due to the special residual connections that allows very deep neural networks to be trained. We used an 18-layer 3D variant of the model as a feature extractor. So once the model is trained, instead of using the complete model for inference, we use the output of the 17th layer as the latent variables representing the ResNet features. So in this way, we use it as a feature extractor to extract 512 features from each ROI. So before we extract any features, we simulated the appearance of bone in clinical CT systems. This is important because the value of texture features are typically very sensitive to image quality. The use of simulation allows us to model the appearance of trabecular bone as visualized in many different imaging systems, from conventional MDCT to micro-CT, which is the current gold standard method to characterize bone microarchitecture. We also simulate as many noise realizations as we need because we're running these simulations. The simulations we use here is based on a mathematical model called cascaded systems. So the sequence of images here in the slide represents the original segmentation of a trabecular bone ROI, which is our ground truth image, and followed by the imaging simulations using micro-CT parameters, then high-resolution CT parameters, and then conventional MDCT parameters. So for the purposes of our work, all of our ROIs were simulated using high-resolution CT parameters, as highlighted by the blue box. These acquisition protocols are not yet what's commonly used for evaluating osteoporosis, but they are becoming increasingly available. As mentioned before, our goal in this work is to predict bone stiffness. To generate the reference stiffness values for training and testing, we applied microfinite element analysis to the ground truth ROIs. Note that this analysis is currently only possible with micro-CT and cannot be performed clinically in vivo. So starting from our data set of micro-CT images of lumbar trabecular bone, we used Otsu segmentation, marching cubes, to convert the image into a volumetric tetrahedral mesh. We then used finite element analysis to simulate a uniaxial compression. Through this, we get the mechanical stiffness of the overall bone structure. And this stiffness value is what we want to estimate from texture features extracted from clinical CT images. So those are images with a lot lower image quality. Our data sets consist of ROIs extracted from micro-CT images of 70 lumbar vertebrae bodies. The ROIs we're starting with is approximately 15.3 millimeters. We applied imaging simulation and microfinite element analysis to all of the ROIs, and then we split the ROIs such that the ROIs derived from the 40 vertebrae are reserved for training, 15 are reserved for testing, and the final 15 are reserved for 15 for validation, and the final 15 for testing. We used a random forest regressor to aggregate the outputs from different combinations of these models into a final mechanical stiffness estimator. We chose random forest because it generally performs well for heterogeneous data and is quite interpretable. The performance for each model was then evaluated with regression factors R squared and root mean square error. So this figure compares the effectiveness of using ResNet, radiomics, and conventional BMD at estimating stiffness, shown here as models 1, 2, and 3. So overall, the image on the left, the ResNet, that's a model based on deep learning, and that shows better performance over the radiomics for our data set, both in terms of lower RMSE and a higher coefficient of determination. So model 3, the model on the right, which is BMD only, that's analogous to the current standard of care. And for our data set, has the worst performance out of the 3. So this suggests that in trabecular bone, there are higher order features that our batch of conventional radiomics features are not capturing, that is only accessible via deep learning approaches. So in conclusion, we compared different feature sets at estimating bone stiffness from texture features. We show here that texture features offer some benefit in estimating bone stiffness from high resolution CT. Now, there are some limitations with these using texture features. As mentioned before, these texture features are very sensitive to changes in image quality, and that's part of the ongoing work. We are looking at that dependence. So some of the factors might include reconstruction kernels, geometry, voxel size, and there are many other aspects of the imaging chain that we could look at. Additionally, to address the lack of high-quality micro-CT data for trabecular bone, we're also building a tool to generate synthetic trabecular bone models to supplement our model training. And we plan to make this model publicly available as a regulatory science tool by the end of this year. So we're looking forward to that. And I'm going to conclude my talk here. Special thanks to my team and collaborators, my colleagues at DITSER, and thank you for your attention.
Thank you, sir. That was an interesting talk on assessment of bone stiffness and how the radiomics and deep learning features can be used to evaluate that. Okay, that concludes our presentation. Then we need to move on to Q&A. And I see a few questions, so I'm going to go over that. And then I'm sure they have identified the speaker that they want to answer the question. Okay, the number one, for Dr. Wu, your strategy appears similar to other therapies that have found a distinct lack of animal model pharmacological relevance, mostly due to the significant differences in immune systems among species. How have you established human safety before your clinical trials?
Yeah, thank you for that question. I think it's exactly the uniqueness of humans as a species that led us to really rely on prior human data. The format that we went after for our vaccines was peptides. Before we came to the agency to start to initiate our trials, we pointed out that, in fact, there were other groups that had already, for example, pooled up to 15 peptides together for tumor-associated antigens, and in published trials had demonstrated that there was a lack of toxicity or adverse events through that approach. So that gave us the confidence that pooling our peptides, again, using formulations that have been previously used in human subjects before would be a safe approach.
Great, thanks. The second question is from Julie. It is also Dr. Wu. She's asking, can you talk more about the current work in identifying new neoantigen sources beyond somatic SNPs and indels?
Yeah, no, thank you for that question, Julie. I think this is an active area of investigation in the field. I think that there are new sequencing technologies that afford us the opportunity, and also the cost savings now as the cost of sequencing approaches like whole genome is momentarily about to come down and be halved for what they are. So I think this affords us the opportunity to really, there's the whole genome sequencing part. There's also long read sequencing that's happening right now that there's uncertainties that happen with the short read sequencing data that now can be overcome with long read sequencing. So I think we are at a time that I think we will be doing better in terms of looking at fusion events or copy number events. Our group also looked at kind of non-canonical open reading frames coming from whole genome sequencing. So these are all areas that are kind of in active testing to both look at modes for identification, but also testing immunogenicity.
Great, thanks. This question is for Dr. Kamal. How is the drug loaded added to the MNs? How is the drug released on the skin? Dr. Kamal, you're muted. You need to unmute, please.
Sorry about that. So thanks for the question. Actually, the drug was added into the casting solution, very simple way, and then applied into the mold. The casting solution was prepared just by adding the various aqueous concentration of the polymers with fixed concentrations of API. And the next question is how is the drug released on the skin? We did not get a chance to look at the drug release into the skin at the sink condition yet. However, when we looked at the drug release under the skin at non-sink conditions that I have presented today, drug release was increasing up to a certain time points by increasing the time points or even by increasing the drug concentration in the needle. But then the release profile after certain points shows steady drug release. It may be due to the limited volume of the moisture present in the skin. Besides, the method was unable to differentiate any change in the formulation variables in the needles. So this is why we couldn't conclude anything from this method. Now we are looking forward to seeing drug release under the skin at sink conditions. Then we'll find out how the release profile is behaving. Thank you.
Thanks. I guess this is from Alex. So it's probably, I don't think it's a question, it's probably a comment. It says there has been studies using microneedle as insulin patch. I hope this will be commercial potential.
Yeah, I hope so.
All right. So the next question is for the last speaker, I think. With regard to, this is from George Burns, with regard to cell therapy-based regenerative medicine, how are guidelines for the potency assays evolving, and what might be done to promote a strategic harmonization and its successful development?
That's a really great question. You know, potency assays can be very challenging for our cell and gene therapy product sponsors. It has been a hot topic, and you know, we encourage our sponsors to test multiple attributes for characterization early in development, with the expectation that you know some assays can be dropped later in development. But you know, there is a 2011 guidance, and you know, hopefully the agency can update the recommendations with knowledge we've gained thus far. But you know, it's very hard because we try not to be too prescriptive when we do interact with our sponsors, because we do want to allow flexibility in assay development. But I do know that there's lots of workshops and lots of different liaison stakeholders holding meetings to try to talk about harmonization, particularly for product classes, assays that may be able to span different products.
Excellent, thanks. And those were the only questions I see. So that concludes session 7, and I would like to take this opportunity to thank all the speakers for their excellent presentation. And according to my count, the 336 attendees for attending this session. Don't forget to check out the posters, and I guess time to say goodbye and have a good afternoon. Take care. Bye.
Thank you, everyone.
Thank you.
Thank you, everyone.
Recording stopped.